Am I audible to you guys? We'll be starting soon.
Yes, you are.
Hello guys, this is Minhal Akbar. Welcome to the third day of IBRO APRC Neurosciences School. This is Minhal Akbar presenting Eric proudly. So we'll start our session today. This school will focus together all the experts in the diverse field of neurosciences working on solving the mysteries of degenerative brain and to discuss this public health burden of neurodegenerative diseases with the recent advances of human and animal research that will help to integrate participants with a growing opportunity for revising pre-existing methodologies, enhance critical thinking about new techniques, and establish them locally. So, proceeding towards this session, I would now introduce the first speaker of the session, Miss Jean King, Dr. Jean King. She is WPI Peterson Family Dean in the School of Art and Sciences. She also serves as a professor of biology and biotechnology, affiliate professor in biomedical engineering department, professor in neuroscience program, and director of neurotech suite at WPI. Dr. King has published over 100 original scientific publications, including papers, book chapters, and review articles in highly respected international scientific journals. The long-term goal of Dr. King research is to provide an understanding of the unique features of central mechanisms that regulate neurobehavioral health and well-being. Her group uses multi-model approach to identify and characterize neural plasticity and behavior modification associated with stress and resilience, with emphasis on focus area including mindfulness, addiction, pollution exposure, ADHD, depression, fearfulness, anxiety, neurological disorders, aging, cognition, pain, and sex differences in model system and human, with the hope of finding clues to help us better understand these conditions and disorders. Her new lab utilizes a transitional approach which incorporates both clinical research and preclinical studies. In 2018, she helped launch the Neuroscience Initiative at WPI, fostering the establishment of a graduate program and interdisciplinary neurotechnology suite at the participant point. At the suite has a number of experienced faculty in area like data sciences, computational biology, EEG, eye tracking, sensors, functional magnetic resonance, functional near-infrared spectroscopy, and physiological monitoring. This multimodal hub support innovative collaboration to advance our understanding of central topics in the neuroscience. Unfortunately, Dr. Jean King could not be available on a live session as due to the time differences. So we will be providing you with the lecture she had considerably provided us before. And for the question answer session, we will be having his research professor, Mr. Ben. So we'll just start with the session.
Thank you so much for joining us today for our presentation entitled Comparative Neuroimaging of the Neurodegenerative and Behavioral Effects of Substance Abuse and Air Pollution. My name is Jean King, and I'm the Peterson Family Dean of Arts and Sciences and a Professor of Biology and Biotechnology at Worcester Polytechnic Institute in Worcester, Massachusetts, USA, along with my colleague, Professor Ben Neveu, who is the Assistant Research Professor of Biology and Biotechnology here at WPI. First, before we start, I would like to thank the Center for Comparative Neuroimaging, where all of the studies we'll report on today were done. On the left is the Center for Comparative Neuroimaging here at WPI. On the right is the Center for Comparative Neuroimaging at UMass Medical School, which is 2 miles away. First up, a quick overview. Today, we'll talk to you a little bit about substance abuse and neurodegeneration. We have clinical and preclinical models. We'll first address the preclinical studies of nicotine. We'll do some clinical studies of nicotine. Then we'll do some preclinical studies of alcohol abuse. Then we'll get to the air pollution work that Professor Neville will present, the background on air pollution and the preclinical studies of particulate air pollution. First of all, the impact of tobacco use and addiction. Over 443,000 Americans, over 18% of all deaths, die because of smoking each year. Secondhand smoke still kills over 50,000 of them. Smoking killed 6.5 million people in 2015 and is set to kill 8.3 million humans in 2030, with the biggest rise in low and middle income countries. Every 6.5 seconds, a current or former smoker dies. Tobacco use will kill 1 billion people in the 21st century if the current smoking trends continue. So why are we beginning with nicotine when we're giving you a talk on neurodegeneration? Well, smoking is highly prevalent across the globe, and nicotine relapse typically occurs within the first month, about 80%. And after 6 months, the success rate is only 3%. Smoking is linked to co-use with other drugs, worsening the brain insults, and possibly leading to more neurodegeneration. Strong correlation between smoking and microstructural brain changes, which we're going to show you today, possibly supporting the development of neurodegenerative disorders. Smoking has a strong risk for cognitive deficits and Alzheimer's disease, especially in individuals without the APOE4 allele. Some of these neurodegenerative effects have been strongly linked to increased neuroinflammation. Now, tobacco use in Southeast Asia, given that this IBRO is in Pakistan, we wanted to take a look at Southeast Asia in particular to look at that versus what's happening in the US. In the US, about 20% of men and women smoke. In Pakistan, what we're seeing is for Pakistani men, it's about 30%, and smokeless tobacco is a little under 30%. I would say probably about 28%. For the women, it's probably less than 2%. So again, the numbers are very high for males, but not as high, thankfully, for women. But there are other countries like Indonesia, when the number of smokers who are male are highly disproportional in terms of the number of people who smoke. Now, smoking is not just about the acute smoking that we worry about. We worry about it when it becomes an addiction. And obviously, addiction is a chronic relapsing condition in which compulsive use persists despite the known negative health outcomes, which means because it's very difficult to stop smoking, some of the neurodegenerative factors, once in effect, gets worse. Now, in our lab, we have used many tools to explore the link between addiction and neurodegeneration, mainly looking at the brain. So, translational neuroimaging is in fact what we focus on. It's a non-invasive approaches to facilitate longitudinal studies in preclinical and human studies, given a bidirectional flow. That's why we do preclinical work and clinical work. The preclinical models support this non-invasive and invasive methodologies. It allows us to do prospective studies. So in the lab, we have really spent a lot of time using both preclinical models and human models of neurodegeneration. Now, in order to study preclinical models, we really had to come up with our own techniques. We spent a lot of time on this, and about 10 years ago, we came up with a way to non-invasively approach the study of awake animal imaging by coming up with this piece of equipment that our lab took a lot of time to come up with. And after 10 years, we were able to do these longitudinal studies. And again, longitudinal studies allow us, especially in preclinical design, to have a prospective analysis of what's happening in terms of brain decay and brain degeneration over time. Now, how do we use preclinical models? Well, there are different ways you could use preclinical models. There is the face validity, and face validity is just a reasonable representation of the fundamental behavior. Whatever the behavior is or the deficit, we'll have an animal model that supports that. Or the predictive validity model, that's a response to drug treatment. So drugs that work in a particular condition works to treat that particular condition in an animal model. For example, our model of ADHD response to Ritalin is one example of that. And third and last, what we call construct validity, which means it conforms to some of the basic mechanisms contributing to the disorder. So we use these preclinical animal models to understand the mechanisms. So any one of these or all three of these could be the approach to a preclinical model. Now, we have a lot of surrogates. And in order to get a surrogate for addiction, we looked at the behavior. And in the animal model we're going to be showing you today is an animal model of a behavioral component of addiction. And in this case, what we did was actually gave the same amount of nicotine to an animal over time. And over time, the same amount of nicotine produced a different effect. This is called sensitization. And sensitization occurs when the subsequent doses, exactly the same dose, produce this greater effect. Now, after a few doses, 5 to 6, we have what we call the expression of sensitization. And in this graph, you'll see that more clearly aligned by day 5 and day 6, the animals given nicotine compared to the animals given saline was doing much more locomotive activity. This is what's referred to as sensitization and the animal model that we'll be showing you. Now, what we are focused on, obviously, because we talked about nicotine, we talked about the addiction circuitry. The addiction circuitry, as we know, composes of the prefrontal cortex and the frontal cortex here, the striatum, the substantia nigra, of course, the nucleus accumbens and the VTA. Of course, reward and motivation, pleasure and euphoria, motor function, compulsion, and perseverance are all functions of the dopamine pathway. So in effect, we're looking at the dopamine pathway in response to nicotine. And what we found was that these preclinical approaches will allow us to sort of design this prospective study and have a way of non-invasively looking over time for this bidirectional flow of information. And here you could see first the analysis in terms of the structures is in A. In B is acute, which means that's the first time the animal has seen nicotine. And you could see if you're just looking at the red that there's activation in these brain regions. But in C, you have the sensitized animal. This is the animal that has been sensitized day 5 or day 6. Then we give them nicotine in the magnet and see the response. And you could see a definitive response. Even if you've never looked at a neuroimaging study before, you could tell that there's much more yellow and red in the figures at the bottom in C as opposed to in B. And what does that really mean? So here is an outline of what happened. In the prefrontal cortex, a region that I told you was involved in addiction, what we found was in the acute, after a couple of minutes, the activation started to decrease. However, in the sensitized, it continued to increase and stay elevated. That pattern is seen throughout most of these regions, including the nucleus accumbens, another region that we know is involved in addiction, the VTA, the ventral pallidum. You'll see an increase in sensitization as opposed to the acute. Looking at the difference between a model for more of the first time use compared to the addicted use. And that's similar in terms of the hippocampus, a region that we're going to talk a lot more about when we talk about memory, the septum, and the anterior cingulate cortex. So what we know is that nicotine was activated in all these regions. So the answer was, well, is it activated globally, every single region in the brain? And what we show you here in terms of the visual cortex, and that's in fact not the truth, that in the visual cortex, it was activated initially, and then it all sort of worked out where there was a plateauing. Whether it was the saline or the nicotine, you got the same response after the initial response. So what do we think about in terms of the preclinical summary here? Here we had seen that this chronic nicotine exposure led to behavioral sensitization, which is our animal model for addiction. It's associated with this increased activity in the hippocampus, in the nucleus accumbens, in the prefrontal cortex, in the VTA, the amygdala, and the ACC. All regions that we talk about that we know is involved in arousal, but also involved in memory. And we're going to get to that. The chronic nicotine did not change in the visual cortex, so it's not a global response. It's a response in selective areas. Neuroplasticity that accompanies sensitization or addiction model and long-term changes may act as a substrate to subvert not just the addictive process, but neurodegeneration, which we'll show you coming up. So in these preclinical studies, is there in any way is it linked to our human studies? Because in the end, we use preclinical models to help us understand the clinical situation of what happens in humans in addiction. So all of these preclinical models, which of course, because they're prospective, we're able to see the amount of nicotine, the level of nicotine, and when it led to an addictive model. In humans, obviously, we have to meet humans where they are. So this allows us to figure out how to actually form a template for the human studies. So what we did was we recruited humans, 20 non-smokers and 20 smokers, ages 18 to 40. Smokers must not smoke at least 11 hours prior to imaging. And then in the magnet, we gave nicotine to people who are addicted to nicotine and see how their responses happened, and we were able to track them. So what you see is bold activity in many of the areas that we just talked about in the preclinical study. Again, showing that the clinical study is very similar in terms of what we got with humans. And the nicotine administrators in the smokers compared to the acute administration to non-smokers. So somebody who got it the first time, similar to the rat study, as opposed to people who got nicotine, who are addicted to it, is associated with increased bold activity in several of these brain areas that we talked about, including the cingulate cortex, the posterior cingulate cortex, and the superior parietal lobe. And chronic nicotine exposure alters bold in selective brain regions in a manner that supports not just the components of the addictive process, more long-term changes in neurodegeneration and cognition. When we looked at the translation, so if you put the preclinical model right alongside with the clinical model and ask what are the differences? Well, several brain regions appear to be exactly the same. Again, the idea is these preclinical models are serving a purpose because the regions are exactly the same. And you could see side by side the prefrontal cortex in rats, the superior frontal cortex in humans, the anterior cingulate cortex in both, insula in both, caudate putamen in both, retrosplenial cortex on the rat side, and the posterior cingulate cortex, the thalamus, and the hippocampus. Now, these brain regions, again, are associated with arousal, reward, sensory integration, and memory. And the activation in response to the nicotine cues in both preclinical and human studies are exactly the same. Now, what are the critical brain regions impacted in nicotine addiction? Now, these are regions that, as you think about it, are regions that we think about in terms of neurodegeneration and Alzheimer's disease. So in the retrosplenial cortex is a critical transit region between the hippocampus and the cingulate cortex. So it takes information flow between those two. It has been implicated in spatial navigation and in memory. More importantly, the retrosplenial cortex atrophy is a prediction of a site for Alzheimer's pathology. Again, because nicotine changes the site and the site is associated with Alzheimer's, we have an opportunity to see if any of these structures that are affected by nicotine are also impacted by Alzheimer's. Another one of those regions is the PCC, the posterior cingulate cortex. It plays a central role in a slow but reliable memory decline in the elderly. Because the functional alterations in these regions are currently described in Alzheimer's disease, this functional signature may constitute a risk for Alzheimer's disease. So again, we're looking at these areas, wondering if after the insult, we could in fact impact the neurodegeneration in these possible areas associated with Alzheimer's. And finally, the hippocampus. The hippocampal atrophy correlates significantly with decline in cognitive status in patients. And it also predicts sometimes a conversion from mild cognitive impairment to Alzheimer's itself. Again, a region that we're really interested in. Besides our studies, others have also shown hippocampal changes. And in this study by Wu et al., right in 2020, just over a year ago, under a year ago, we find that on the left side, the changes in glucose, that there's a decrease in the smokers as opposed to the non-smokers. And they also, in terms of the volume of the hippocampus, a decrease in the volume of the hippocampus in smokers versus non-smokers. Again, linked to the nicotine studies, where we're showing these changes in the hippocampus. In human studies, there are also changes in the hippocampus in smoking, and this will more likely aligned with Alzheimer's. In fact, other studies have shown the association, where the cigarette smoking is cerebral spinal fluid of neurodegeneration and neuroinflammatory markers and oxidation markers. In this study by Liu in 2020, what you see in A is the beta amyloid is increased, as we know in terms of Alzheimer's, this is a marker for Alzheimer's, is increased in the active smokers compared to the non-smokers. Similarly, TNF alpha is increased in the active smokers compared to the non-smokers. And TNF and BNF actually is in the opposite direction. It's decreased in the active smokers as opposed to the non-smokers. In nitric oxide and oxidation, there's not as clear story as they are in terms of the amyloid, beta amyloid. Again, proving that there's an association or a link between cigarette smoking and some of these markers of neurodegeneration. This also we show in other studies. Researchers have been showing, like Wu et al. In this study shows that there's an association between memory decline in normal individuals, normal older individuals, in ways that might suggest that it's leading to some sort of cognitive deficits, especially in these mini deficits in smoker status, in a mini mental state in A, and in C, the total learning score is different in smoking, with smoking less than the non-smokers. So in A and C, there are cognitive changes that are significant. So what do we think about as we think about these structural changes? I just showed you that they were activation changes, which may mean they're functional changes. But are there really changes in terms of the psychoarchitecture of the brain? That's a different question. Did the brain change in terms of the psychoarchitecture in people who smoked versus in people who didn't smoke? And what does that mean in terms of how much you smoke, whether the brain architecture changed depending on how much you smoked? So what we're able to show so far in terms of our studies and others is that nicotine changes the brain. And it may change the brain in a direction that's leading to more of a pathology towards neurodegeneration than in the opposite direction. So what we do, because we have a comparative neuroimaging lab, we have a lot of different neuroimaging techniques, but the one that tells us about the structure and the microstructure is called DTI. And DTI is diffusion tensor imaging. And it's a method that demonstrates the orientation and the integrity of the white matter tracks in vivo. Again, this is a non-invasive approach, so we're able to look for these tracks in vivo. Within the cerebral white matter, water molecules tend to diffuse and are more freely along the direction of the axons than across them. Such direction dependence of diffusivity is termed anisotropy, and we're going to show you a little bit about how that changes in terms of smoking and non-smoking. So what we found this in terms of FA, which is fractional anisotropy, of the left anterior cingulum bundle, which we talked a little bit about in smokers, is different from non-smokers. So you see the left side and the right side. In the left ACB in smokers, there's a big difference in FA compared to non-smokers. Similar in the right, there is a big difference between smokers and non-smokers. So what it does show is that in fact these microstructures, as you could see in your pictures, is that red zone is different whether you're a smoker or a non-smoker. This change actually is associated with different projects in terms of what we did in terms of addiction. And this was really important to us. So the FTND, which is something that physicians use to tell how addicted somebody is to cigarettes, and the kinds of questions that are asked on the survey is how soon after you wake up you smoke your first cigarette, how many cigarettes you smoke per day, do you smoke if you're ill and in bed most of the day? These kinds of questions produce an answer and a score, and that score is called the FTND score. And what we found is that smokers in that left ACB that we just talked to you about negatively correlated with the FTND. So the more that the smoker was addicted, the difference in that line decreased. So there was an inverse relationship between how much you are addicted and how much you had changes in terms of the FA in these regions. Telling you again that FTND, the addiction value and what the changes in the brain were correlated. So after doing FTND, which is the standard, we really wanted to look to make sure that this one behavioral paradigm didn't just pick up something that was odd. So we also administered another questionnaire about how addicted a person is, and that's called a hunk. And this one sort of stresses more about cravings than about the number of cigarettes. So you could see questions here Or like, do you ever have strong cravings to smoke? Did you feel more irritable because you couldn't smoke? So it's much about the emotional component of smoking as opposed to the quantity of smoking. And what we showed is similarly, with the hunk, there was this correlation between how addicted you are and the brain changes. And this was really important to us to show that not just one of these values, behavioral questionnaires showed this, but this other one, the hunk. And then we did what was the third way of looking at addiction, what we call the physical dependence model, which was developed at UMass Medical School. And this was basically about how you respond to smoking. So questions like, if I go too long without smoking, I just can't function. So it ties the amount of cigarettes with the functionality of the person. I know I have to smoke just to feel normal again. If you say yes to that, you're at level 1. If no, you continue on this. And what the next question is if I go too long without smoking, the desire for a cigarette becomes so strong that it's hard to ignore and interrupts my thinking. If you say yes, you're at level 2. If you say no, you continue. And of course, the last one is if I go too long without smoking, the first thing I notice is a mild desire to smoke that I can't ignore. And that's the level 0. And when we looked at these PD values, of course, what we found is what we had found before in the 2 other behavioral markers. That with physical dependence, depending on how dependent a person was, the change in the brain slope was exactly the same. So there was a definite correlation in smokers between changes in the brain that was related to how dependent they were on nicotine. That was quite a surprise to us because this was convergent evidence from 3 different measures of tobacco dependence that supported the association with the structural integrity of the brain. That these microstructural brain alterations set the course for what we think could lead to neurodegeneration. Now, smoking, dementia, obviously Alzheimer's and APOE4. Smoking was associated with doubling the risk of dementia, and this was a study by Otter in 1998, where smoking was associated with doubling of the risk of dementia and Alzheimer's disease, where carriers of the APOE4 had no increased risk of dementia, suggesting an interaction between smoking and APOE4 genotype in the etiology of Alzheimer's disease. The reason we are reminding you of this study is because there is some discrepancy in the literature where people say smoking is protective and other people say smoking is not. So what we want to remind you of the study that says it's probably the genetic predisposition that's actually fused with and compromised by the use of nicotine. So in some cases, you'll see these definite patterns, and in others, you probably won't. And that's probably why there's discrepancy in the literature, because very few studies actually did the ApoE correlations. So it might be in many other studies where we just haven't picked it up before because we haven't done that. So we wanted to bring you back to that Lancet study. Next up, outside of nicotine, we wanted to show that in fact, these insults are not just about nicotine, that other substances of abuse could also change the structure of the brain in different ways. But why is alcohol important? Again, 1 to 5% of the population suffers from an alcohol use disorder. That's a large population. Alcohol use disorder is associated with increased risk of dementia, Alzheimer's, and Parkinson's disease. So again, the use of a substance tied to neurodegeneration. And these adverse neurodegenerative effects of alcohol use disorders are strongly linked, we think, to oxidative stress and inflammation, and we'll show you a little bit about that. So in the first case of nicotine, we didn't show a mechanism. Really what we showed is there's an association between nicotine, changes in microstructure that might be able to predict how Alzheimer's could develop. In this case, what we're talking about is a mechanism behind alcohol's use and dementia and Alzheimer's. So in our lab, we showed in an animal model, in a preclinical model, that chronic alcohol-induced memory deficits. So you had the pear-fed and the alcohol-fed animals. And what you see here in this spatial memory object location, which is a test that we do with preclinical animals to show that they could remember where an object was previously paired. And after you move that object, they would return to that space. And what we showed is the pear-fed remembered where the object was as opposed to the ethanol-fed, which showed a deficit in finding the object, which showed a change in memory. and a memory deficit. That was definitely in the alcohol group compared to the non-alcohol group. And this chronic alcohol, the same model, was used to look at brain factors. And basically at this time, looking at a reduction in the brain in several areas of supporting oxidative stress in terms of glutamate and NAAG and inositol and choline. You see in all of these, the alcohol changes due to alcohol. That alcohol, in fact, affected several of these markers of oxidative stress. And that it increased neuroinflammation and neuroinflammatory markers. And again, here we did two areas, the dorsal hippocampus and the prefrontal cortex. Again, if you remember, these are areas that are involved in addiction. These are areas that we show changes in nicotine, and these are areas that we know are involved in memory. Again, the same here. These areas involved in memory and involved in addiction changed in response to alcohol as compared to the non-alcohol groups. And the PFED, so we have TNF alpha changes in terms of the mRNA. We have IL-6 and IL-1 beta. All of these neuroinflammatory markers in terms of the mRNA level changed, and you see an increase in terms of the ethanol fed versus the PFED. Again, showing that neuroinflammation in the brain is accompanying alcohol exposure. And the liver markers, or similarly, we showed that this was leading to liver injury, that these markers were the same in terms of spatial memory. And again, showing that the liver and the brain were really connected in this area, and we are really trying to figure out how to connect the two. And this alcohol, so basically what we're saying here is neurodegeneration in terms of alcohol and Alzheimer's may be linked through these neuroinflammatory pathways. So we have the peripheral, we have the consumption. Once it gets into the brain, all of these neuroinflammatory pathways are in fact induced and accelerated, leading to what we think as a process for neurodegeneration through neuroinflammation. Next up, my colleague, Professor Ben Neveu, will talk to you a little bit about the impact of air pollution in the same vein that we were talking about before. Ben?
Thanks, Gene. Thanks. Yeah, so a few years ago, Gene and I collaborated with some other investigators at Tufts University that had no background in animal research. They were epidemiologists looking at the adverse effects of air pollution, which is common across the world. In Pakistan, at least 10% of deaths are attributed to air pollution. There's been estimates as high as 20% of deaths linked to air pollution in Pakistan. And we have similar issues in metropolitan areas here in the United States. There have been strong associations from epidemiological data between particular air pollution, dementia, as well as Alzheimer's and autism. So the project that we were involved in was looking at the links between gestational and early life exposure to air pollution and increased rates of autism. So researchers at Harvard have published data on epidemiological findings, but there wasn't a lot of animal work done in this area. So the adverse neurodegenerative effects of air pollution have been linked in other animal models by increased systemic and neuroinflammation, but the data in this area is pretty limited. Thanks. So this is just showing you an overview of the global impact of air pollution and its attribution to particularly particulate air pollution and its link to increased deaths across the world. As you can see a lot of the Asian countries have very high levels, north of 9 or 10%, including Pakistan. Next slide, please. So why are we interested, and what does particulate air pollution do in terms of its effects on the brain and behavior? So you see high levels of air pollution around transportation hubs, areas of high population density in general, due to increased use of fuels and factories and industry, and also areas with indoor pollution. So whenever you have indoor cooking, it's a pretty potent source of particulate matter air pollution. Particulate matter accumulates in different brain regions. There's some amazing work some of the critical work in this area is done in Mexico City compared with areas that have much lower levels of pollution. It causes epigenetic changes both in periphery and brain tissue, and robustly induces inflammation both in the periphery and in the brain. So how does it do this? Particulate matter, small particulate matter, either fine or ultrafine, can directly access neural tissue through crossing the blood-brain barrier. It can also damage the blood-brain barrier, and typically cannot be eliminated through phagocytosis. So people have actually documented individual particles of particulate matter in the brains of both dogs and children in Mexico City and other cities and countries around the world. So what this ends up doing is causing chronic neuroinflammation, and these effects over long term have been linked to adverse mental health outcomes related to neurodegenerative effects. Next slide. Thanks. So impacts of particulate matter on mental health particularly. So our interest was in looking at the combined effects during gestation as well as early development. But what they've seen in both animals and humans is systemic and brain inflammation, and they've linked that to neural fiber changes, for example, changes in DTI, and then behaviorally memory deficits and increased incidence of Alzheimer's, the risk of Alzheimer's is strongly linked to even dose-dependent link to particulate matter levels. Next slide, please. Thanks. So our project was involved in looking at genuine Boston area pollution that was collected in one of the traffic tunnels. And our hypothesis was that developmental exposure to TRAP, or traffic-related air pollution, will induce systemic increases in inflammatory markers, neuroanatomical changes, and behavioral and cognitive deficits similar to children with autism spectrum disorder. So that was our hypotheses. And how we did this was, we can go to the next slide. So this gives you a little snapshot of our setup here, which was done by engineering students at Tufts previous to my coming to WPI. So we exposed rats, so maternal rats, to trap collected from a tunnel in Boston, or ambient air from our animal facility for 5 hours a day, 5 days a week, for 6 weeks during gestation, which in rats is 3 weeks, and then another 3 weeks during lactation. So this is basically all the critical periods of early development. And then we were looking at their behavior when these animals were juveniles. And due to the increased incidence, much higher incidence of autism in males, this study was just focused on male juvenile offspring. Excellent. So what we saw was changes in behavior as well as brain morphology. So we saw a decrease in allogrooming, which is what juvenile animals do when they're together, and it's very important for the development of social behavior, leading to eventual, normal, typical displays of sexual behavior, as well as parental behavior when they're older. So they groom each other when they're in groups in cages together. The pollution-exposed animals had a significant decrease in this, and they also play. So they run around, they chase each other, they wrestle, and we saw lower levels of this social play, which again is very important for the development of adult social behaviors. And we saw, a significant decrease in the pollution-exposed animals. So neuroanatomically, as Jean introduced DTI analysis, we were able to do ex vivo DTI on the brains of these control and pollution-exposed animals, and we saw decreased FA, or fractional anisotropy, in the hippocampus as well as the anterior cingulate, and this was likely to be involved in some of the behavioral changes we saw, but the sample sizes in this particular experiment were too low to actually allow us to do a correlational analysis. Next slide. So the summary from this experiment was that chronic exposure of rat dams and their offspring to trap during gestation and lactation resulted in decreased social behavior, increased anxiety, impairs cognition. We had some cognitive tests done on these animals and we saw impairments there. We actually saw decreased levels of inflammatory factors. This is a complicated story because we were only able to measure peripheral levels, but the decrease we saw is often associated with an increase in central levels of this particular inflammatory factor. So that was interesting. And we saw changes in growth factors, which you actually see similarly with the smoking. And these were correlated with behavioral changes that we saw. And then we also documented the disruption in neural integrity through the DTI imaging. So exposure to traffic-related air pollution during gestation and lactation, in summary, it's likely to be involved in the etiology of autism spectrum disorders and other disorders. There's other disorders which involve social and cognitive deficits and or increased anxiety. So this goes beyond autism to other disorders that involve neurodegenerative changes in the brain.
So overall, we're talking about these comparative multimodal neuroimaging is really effective at identifying and characterizing translationally relevant neurodegenerative disease etiology. It allows us in the preclinical models to look at timelines in a prospective manner. So we're really interested in using that. We only talked to you today mostly about resting state functional changes, and we talked to you about DTI, but we also use MRS, which tells us a little bit about the chemistry. So the lab, the multimodal approaches is both in terms of function, chemistry, and anatomy. So we do get a composite of looking through in a comparative way across these multimodal approaches. The insights from this can be used to inform the development, we hope, of preventive measures and treatments to inform policymaking. The neurobehavioral and neurodegenerative effects of substance abuse and air pollution in rodent models and humans are highly conserved. Obviously, you've seen it between species. It turns out to be, in fact, the same sort of brain areas that are impacted, whether in animals or in humans. We think a lot of this might be linked to neuroinflammation as a process to get to neurodegeneration. We have shown you in some instances that we have seen changes in neuroinflammatory markers. We're doing more to sort of expand that, to think about what are these neuroinflammatory markers and how they induce neurodegeneration. We know there's a process and a line towards that, as you're seeing in this picture, that dementia, stroke lead into these sort of acute neuroinflammatory responses that then become chronic and then lead to changes that, again, in this loop back, goes into the microglia and astrocytes impact in terms of being ready to help the neuron if the neuron is being impacted. And again, if it's being impacted in a way that is not being able to survive, it leads to neurodegeneration. So we think, in fact, the neuroinflammatory markers may, in some cases, tell us what's happening in terms of mechanisms towards neurodegeneration that we observe in a more long-term and chronic manner. We have left you with some resources in terms of the papers that we mentioned. We know this is being taped so that you'll be able to go back online and look at these resources if you're looking for some of the papers that we talked about here today. We'd like to make sure that we give you a list of things that you could get in terms of getting more information. I really cannot do this work, and we cannot do this work without the lab, and we want to thank all of the students and the research technicians, coordination, data analysis, research intern in our lab. We're really excited and happy to have them along to do more of this work as we continue. The Neurotech Suite is a larger suite that we participate in. Both Ben and I are part of this Neurotech Suite, where we have many of the resources we just talked about. We just expanded in terms of the multimodal imaging to do FNIRS and bring online eye tracking and different sort of neurotech approaches to bring in these ideas that we talked about to you today more in a comprehensive way with clinicians and researchers across our school. If you have any questions, they will be addressed online. We're hoping that although we may not be physically in there with you in person, although I think Dr. Neville will be there for the physical talk and be able to answer your questions online. We want to thank you for your time. I really want to thank all of the organizers for inviting us to speak. We're really excited about working with the IBRO School in Pakistan. We look forward to hearing more about the neurodegeneration processes and all of the people you brought together. Thank you so much for the invitation, and we look forward to working with you in the future. Thank you.
Thank you.
Okay, it was a really good talk, an exceptional talk from Dr. Dream. Now the question answer session would be continued by Benjamin Nephew, Dr. Benjamin Nephew, who is an assistant research professor, biology and biotechnology. He has been doing research broadly with a focus of identifying and studying neurobehavioral mechanisms of mental illness and developing effective interventions. They use functional MRI to identify neural correlates of mindfulness-based stress reduction as part of the stage 2 randomized control trials. The functional MRI data combined with related clinical measures to develop machine learning-based early predictors of severe depression and suicidality. They developed a transgenerational social stress-based rodent model of postpartum depression, anxiety, and related clinical work explores the role of behavioral hormones, discriminations, and epigenetic changes in postpartum depression and anxiety. They are continuing animal research includes the impact of social stress, alcohol, and air pollution on behavior, immune factors, neural connectivity, neuroendocrinology, and related epigenetic changes. They're also using functional MRI to study sex and age-related differences in cognition and primates and developing the use of functional ultrasounds to study pain-related mechanism and interventions. They have been doing projects involving integrative collaboration with a range of schools at WPI, UMass Medical School, Harvard, and other institutions. They're looking forward to build additional connections with interested students and faculty at WPI. So we are off to you, Dr. Ben, for the question answer session. So welcome in the conference.
Yeah, thanks for the invitation and for the introduction. Yeah, we appreciate the invite, and it was a great opportunity. And yeah, I'd be happy to take any questions.
Yeah. So the first question that we got from the participants is, which brain part is involved in controlling human behavior in nicotine addiction?
Yeah, so you know, with a lot of addictive processes and etiology, the reward pathways are typically involved pretty heavily. But that's also, so things like the ventral tegmental area and the nucleus accumbens are key areas there. But you know, you also have the involvement of the prefrontal cortex, you know, on a daily basis in terms of how addiction is affecting your everyday decision making.
Okay, that is a productive explanation. The next question that we have is, other than BDNF and TNF, which other cerebrospinal fluid biomarkers are associated with cigarette smoking?
Yeah, so that's not my area of expertise, that's genes. But, you know, I think that with a lot of these inflammatory studies, there's a bit of a bandwagon effect where people just tend to focus on, you know, one's identified and people tend to focus on that. And now that you're able to do multiplex assays pretty easily, I mean, I think it's kind of like when they started analyzing the compounds found in, you know, cigarette smoke. It organizes a whole host of immune factors, or it induces a whole host of immune factors. But I know that, you know, in both air pollution and smoking to some degree, there's been a lot of recent excitement over immune modulator that really seems to direct a lot of immune responses, and that's called NLRP3. And so we're hoping to actually focus on that more in our air pollution studies. but it's involved in a lot of chronic inflammatory responses.
Okay.
I do see there's another question in the chat. As you discussed, air traffic
So
pollution.
the question is from Shuja Heather. He is asking about the discussion of the air traffic pollution. As you said, it is involved in autism spectrum disorder during gestation period. Can you please identify the particular medical sector which causes such neurodegenerations?
Yeah, so that's a great question, and we've been actually working on that recently. So my group does not specialize in immunohistochemistry, so we're hoping to collaborate with others. So we still have additional brains from that study, and we do have someone at Worcester Polytech that does immunohistochemistry, but we need someone with the specific antibodies or stains to look at things like metal. For your question about lead, except specifically, it's, although heavy metals are particularly destructive to brain tissue, and that's well known from water as well as air pollution. To some degree, it doesn't matter like what the particle is, because these particles are so small and they're just foreign material. So no matter what it is, it'll stimulate an adverse immune response. But I mentioned in the talk some of the amazing research done by Lillian Calderon in Mexico City, and they have photomicrographs of magnetite particles in the olfactory nerve of both, I think it's both children and dogs living in Mexico City, and it's really kind of fascinating. It was a major paper, but when I actually reviewed that before we had the ex vivo brains to do the DTI analysis with, I actually called Jeannie's lab. So at that time I was at Tufts and I was collaborating with her when she was still at UMass, and we had to check into if these magnetite particles would just rip out of the brain when we put them in the MRI, because they are magnetized, but apparently they're so small that they don't have enough a strong enough magnetic field to put a problem in a 3T magnet. But it was kind of a funny conversation to have. But yeah.
Okay, that was a good answer to the question. So the next question we have is from Asia Afzal. She's asking, can chronic stress-induced neuroinflammation leads towards dementia?
Yeah, so a lot of my rodent work is actually focused on chronic social stress in mothers and in offspring, and you actually see a lot of the same changes in inflammatory factors in neural tissue. There's stresses are different, but to some degree, if it does alter the immune system, you're going to see similar changes. People, for example, pollution is associated with increased dementia, but chronic depression is also associated with increased risk for dementia and Alzheimer's. So there's a lot of similarities in terms of types of chronic stress and how they affect the brain.
Okay, so the next question is, you have mentioned that hippocampal volume found to be decreased in smokers. Can you please tell us about such changes in depressive people and one with cognitive impairments?
Yeah, so you end up with decreases in things like brain-derived neurotrophic factor, chronic decreases, and that affects microglial activity and will eventually lead to increased atrophy and just more apoptosis than you're going to have in someone that's either not depressed, or a smoker, or exposed to some sort of chronic stress. And eventually, those decreases in either gray matter, so centers of neurons, or the white matter connecting those brain regions, either that's going to affect the the functional activity of key brain regions, or the communication between those brain regions. And that's been, a big focus lately. There's been an increased focus on DTI and structural connectivity, and how that affects the overall functioning of networks that require, very carefully coordinated activity, to be efficient and productive.
Okay, so the next question that we have is, how badly can alcohol-induced oxidative stress affects young adult nervous system?
No, that's a great question, and it gives us a chance to talk about. I actually meant to send Jean a paper that we had just talked about to possibly mention during the talk. So chronic alcoholism is obviously very bad, as well as binge drinking, and you end up with, even acutely extreme rates of oxidative stress with binge drinking. So a lot of alcohol at a single time. And so that tends to be, when comparing the two, even worse if it's, chronic binge drinking. And fascinatingly, there was just a recent article where, kind of, not necessarily just building off our research, but, many other people's research on investigative research on the role of inflammation in addiction in animal models as well as humans. There was an intervention, a clinical trial of an anti-inflammatory for alcoholism, and it had no effect on overall amount of drinks taken in general, but when they looked at binge drinking bouts, that had a very significant effect specifically on binge drinking, which is kind of fascinating that it had such a targeted effect on binge drinking, which, may be very useful for people. And it's just an interesting, intermediate finding. It would have been great if it was effective, like overall at decreasing alcohol intake, but it did have a pretty significant effect specifically on binge drinking. But yeah, with adults' nervous system, so alcohol is cytotoxic in general. So it's just going to cause apoptosis in the brain. And then chronically, it's going to cause, issues with overall increase in oxidative stress. That's going to lead to additional apoptosis as well.
Okay, so Dr. Ben, we have another question which says, are there any ways neurodegeneration can be delayed these days, as we see doctor prescribing ginkgo and turmeric supplements? Is there any evidence that these supplements work for neurodegenerations?
Yeah, no. So that's interesting. What so we actually work with some investigators that do acupuncture and traditional Chinese medicine at the Mass College of Pharmacy and Health Sciences, and they study these things. And then I don't know if any of you are aware, but in the US, there was this latest controversy with an Alzheimer's drug that did okay in a first trial and then had no significant effects in a second trial, but the FDA approved it. It just goes to show, like, to some degree, how desperate we are for any sort of interventions for neurodegenerative diseases, particularly Alzheimer's. I think honestly, the strongest evidence right now is probably general lifestyle changes, not, cutting out excessive drinking, not smoking, and there was just actually a large, I don't know if it was a systematic review or a meta-analysis on, lifestyle things like increasing, maintaining physical activity and neural activity and with, cognitively challenging games throughout later life. I think that's probably where the strongest evidence is so far. But yeah, there's also a lot of research on, so turmeric is a very potent anti-inflammatory. But the trials on this in humans, the data from humans is not particularly strong. with those types of nutritional supplements. I would say the data on turmeric is probably the strongest, if I had to guess.
Right. Dr. Ban, can you please specify to what extent can we delay the neurodegeneration with respect to the pharmacological view or the clinical researches we are yet having?
Yeah, so there is some, Jean didn't present it, but, there is some evidence that, some of these anti-inflammatory treatments may be effective at decreasing the adverse impacts, but there haven't been a lot of clinical trials in this area with regards to serious neurodegenerative diseases. But some of her alcohol research that she did not present, we actually never published, but there is some data showing that if you can block the inflammatory response in the liver, so there's this liver-brain axis with alcoholism, and if you can prevent this adverse response by the liver, you can prevent the cognitive deficits, at least in animal models. But that hasn't been scaled up to clinical trials at this point.
Okay, thank you very much, Dr. Ben. There were sufficient answers and advocate information for us to attain the idea. We'll proceed to Dr. Sadath now.
Thanks.
Hi, Dr. Ben. Hi, Dr. Ben. Thank you so much for all the efforts. And we are very much thankful on the behalf of organizing committee. I think it must be very late night there, and we are very much thankful for all the efforts you and Dr. Jean has made. And it was an 11th hour invitation for the talk, but you guys were the best suited for this talk, and we wanted the whole Asia Pacific region to just connect with this group of scientists. So thank you so much for all the.
Yeah, no, it was our pleasure. It was, yeah, we thought it was a great fit, for our research as well. And again, thanks for the invite, and feel free to reach out to me or Jean if you guys have any questions or are interested in our research, and including collaboration. Thanks.
Thank you so much from Pakistan, and goodbye. Thank you. Welcome, Dr. Lee.
Hello, hi, good morning, good afternoon, and probably good evening to some of us.
Yes, Dr. Lee, we will be just starting the session, and with your introduction, we'll be playing the talk, and then after that, we'll be taking the question answer session.
Okay, sure. Thank you. Thank you.
Thank you so much for giving us the
Hi, I'm sorry, it was on mute. So we would be proceeding with the next session, which would be conducted by Dr. Yang Wiley. He has done his PhD as a gastroenterology fellowship and completed the PhD in Glasgow, United Kingdom, and later a postdoctoral fellowship in United States. He has published his high-impact journal, including Gastroenterology and Gut, a number of book chapters, and numerous conference proceedings. He was awarded the Outstanding Young Malaysian by JCI Malaysia in 2015 and the top research scientist of Malaysia by the Academy of Sciences Malaysia in 2018. Currently, Dr. Yang serving as a professor of medicine and consultant of gastroenterology, hepatology, and internal medicines in University Saint Malaysia. Among his research interests includes the gut-brain axis, gut microbiota, neurogastroenterology, and mortality. Over to you, Dr. Yang.
Hello, and thank you so much for the very kind introduction. I'd like to also thank Dr. Sadaf, who has kindly invited me to speak on this interesting topic, the relationship between gut microbiota, the gut itself, the brain axis, and neurodegenerative disorders. So this is my outline of the presentation. I will start with what is the gut-brain axis, followed by the gut microbiota. And then I will go on to the disorders of the gut-brain interaction, or commonly known in our field as the functional GI disorders. Then I will talk about neurodegenerative disorders, and lastly, the long COVID-19 syndrome. I would like to start with a case study. This is a 52 years old female with a BMI of 39, obviously obese, and she has a diabetes mellitus and also Helicobacter pylori gastritis. And since young, she has intermittent abdominal discomfort, constipation, and bloating, and her doctor diagnosed her with irritable bowel syndrome. At the age of 48, she developed a progressive hand tremors, rigidity, and upon investigation, she was diagnosed to have a Parkinson's disease. Unfortunately, last year in April 2020, her GI symptoms somehow worsened, presented with diarrhea and nausea, and she turned out to have a COVID-19. The GI symptom itself have recovered, but somehow persists, and her Parkinson motor symptoms seem to be fluctuating, even though she's is on a levodopa. So the big question is there a connection between all of this? Let me talk about, first of all, what exactly is the gut-brain axis? It is really a complex, a bi-directional communication that exists between the central nervous system and the enteric nervous system. And obviously, this connection is vital for the homeostasis. I think for those who did not know it, the gut is really the second largest brain after the central nervous system. And the axis itself is made up of a number of components or scaffolds, and this includes the central nervous system itself as the largest coordinator, as well as a neuroendocrine, neuroimmune, autonomic nerve, the enteric nervous system, as well as the gut microbiota. Now, it's not difficult to imagine why they are connected, because they actually share a common embryonic basis. If we were to look at this particular diagram, by week 12 itself, in this 10-week-old fetus, the neural crest, or the so-called central nervous system cells, already migrate to the wall of the gut to form the enteric neural nervous system. And they share the same neurotransmitters and receptors, although the receptors may depend on the location. For example, the serotonin in the central nervous system may be treating depression, but the effect in the gut may actually causes diarrhea. So the effects of of the receptors or the neurotransmitters on the receptors may differ between central nervous system and the enteric nervous system. Say, for example, with the use of a SERT or serotonin epinephrine receptors transporters, it may have an anti-depressant effect on the central nervous system, but in the enteric nervous system, it causes nausea and diarrhea. For the norepinephrine, it may have an antidepressing effect in the central nervous system, but in the enteric nervous system, it may cause dry mouth, sweating, and constipation. Whereas the dopamine, it may increase activation in the central nervous system, but in the enteric nervous system, it may cause nausea. So 5-HT1, for example, buspirone, which causes the 5-HT1 agonism, would have caused antidepressant in the central nervous system, but in the enteric nervous system can cause nausea, headache, and nervousness. And for 5-HT3, with the use of receptor antagonists, for example, mirtazapine, which we all know is a commonly used drug to treat depression, it may cause less nausea or diarrhea and constipation side effects. So it is important to understand also, in addition to the neurotransmitters, what would be the cut to the brain in form of the sensory signaling, especially with the afferent signaling. So because of the gut itself being a viscera or organ, there are visceral efferents which upon the stretch or chemical stimuli, which obviously may depend on whether it is a physiological stimuli to a more noxious stimuli, may then provide a stimulus to the band of endings, which can be found in the mesenteric serosa or even the mucosa itself. And some of these so-called efferent may be silent, or what we term as a sleeping nociceptors that may awaken when in response to injury. Also, some of these receptors may actually be luminal sensing. Some of these vagal or even pelvic efferents may never penetrate the lumen, but it actually senses the chemicals that are absorbed across the mucosa or even from the endocrine or EC cells. And these luminal sensing receptors or efferents may actually have important digestive function. And some of these chemicals may include the cholecystokinin and the GLP-1. And some of these sensory afferent a disturbance may result in two commonly described sensations, including allodynia, a stimuli that normally are physiological but causes pain, and hyperalgesia, which are responses that are actually painful, become more exaggerated. So this comes to a term what we call as the peripheral sensitization, and essentially in the peripheral sensitization, you all started with a peripheral factors, including inflammation, ischemia, or other tissue injury, and this will then produces all these inflammatory mediators, the immune cells, as well as even the bacterial product itself, and all of this can activate the receptors, including the commonly known TRPV1 on the sensory nerve terminals, and this then causes a sensitization, which is basically a form of neuroplasticity. This is the reason for some of these commonly GI symptoms in the in those patients of functional GI disorders, which I will describe later on in detail. But this is typically what we term as the visceral hypersensitivity. It usually results in a very rapid response, but short-lived, although sometimes it can be prolonged if there is a persistent injury through changes in the gene expression. The other sensitization is a central sensitization, and obviously this occurs at the central nervous system level, and this is often chronic and prolonged, and that's why it requires a modification in the genes, allowing modification in the expression of receptors, channels, mediators, and neurotransmitters. This then result in abnormal signaling in the spinal cord. Usually there is a reduction in the descending inhibitory modulation. And because of all of this abnormal signaling, it may actually cause a remodeling of the synaptic connections in the brain. This is what we term as central neuroplasticity. And because of this mechanism, it may explain some of these very chronic GI symptoms that we may see in certain GI disorders. Now, let us go to now the enteric nervous system. And similar to the central nervous system, the so-called ENS is organized into a hierarchical fashion. And this is important to determine the moment-to-moment GI function, because we all know that the gut never stop in moving and also other performing other GI functions. In fact, the ENS is a highly integrated and highly coordinated digestive states, and you can imagine the ENS organized in the form of an app, or all these term mobile apps, and it has like a set of a program library which can be turned on or turned off depending on the situation, as in illustrated in this diagram. Say, for example, if you have acute gastroenteritis because we ate some wrong food, this may actually activate this APRC or propulsion program or application, and also resulting in expulsion of the stimuli, the noxious stimuli, which obviously is a defense program for the gut. And similar to also the central nervous system, there is a chemical synaptic connection between the motor neurons, the interneurons, as well as the sensory neurons, where the interneurons serve to process, to modulate, as well as to execute the application. At the heart of the enteric nervous systems are the so-called interstitial cells of Cajal. These are basically pacemaker cells, as illustrated in this diagram over here. They produce electrical snow wave, resulting in a phasic contractile tension, resulting in the synchrony of whatever program that has been activated. And the interstitial cells of Cajal connect one another to form very active electrical syncytial network in the form of a slow wave that spreads passively through the voltage-dependent calcium channel, then resulting in contraction of the smooth muscles. And some of the disorders associated with loss of interstitial cells of Cajal, for example, Hirschsprung disease, achalasia, diabetic gastroenteropathy, and even slow transit constipation, all of these conditions will result in abnormal or dysmotility of the GI tract. Basically, at the level of the gut wall, there are really two main forms of neuropathophysiology associated with the ENS. First of all, at the mucosal level, this is where the serotonin signaling plays an important role. Now, in this case, where if there is production of 5-HT from the so-called enterochromaffin cells or endocrine cells, this may be terminated by the SERT or the receptor transporter at the basal surface. Now, in the presence of, like, for example, inflammatory bowel disease and IBS, it can be reduced, or even if the SERT is inhibited by the SSRI, then it may actually result in a dysfunction. Now, this can happen when there is a presence of pro-inflammatory cytokines or even a result of gene polymorphisms. The second level is the myenteric plexus, where it plays two important roles. One is the secretion and the other one is motility. Now, in terms of secretion, it's probably much more simple, where it's just 2 neuron reflex circuits. But the motility part is more complex, where it may produce an excitatory signal to the upstream, but it may be inhibitory signal downstream. And sometimes it may, if there is a dysfunction, it may actually result in pseudo obstruction. Another important component of the gut is really the crosstalk between the nervous system and the immune system. I think this is something more has been discovered more recently. Now, with the inflammatory induced neuroplasticities and sometimes persist even beyond recovery. For example, the post-COVID-19 and so on. And as I mentioned, this crosstalk is certainly only known more recently, where it was found that receptors do exist on both the immune cells as well as the nerve cells, allowing a bidirectional communication. So with the activation of the vagal nerve, it may have an anti-inflammatory effects mediated through the macrophages, and it's mainly the cholinergic neurons. Whereas if you have activation of the sympathetic nerve, which may have a differential effect, where you have a pro-inflammatory effect if you have activated the alpha-2 adrenergic, but it may have an anti-inflammatory effect if you have if there is a involvement of the beta-3 adrenergic. Also, the importantly, what really cause the interaction between the luminal environment and the enteric nervous system, and this is really where the so-called intestinal barrier lies. And the intestinal barrier itself, or the gut barrier itself, consists of the epithelial cells, but there are also a generous amount of immune cells in the lamina propria itself. And the two important stimuli that really interact with the gut barrier and the gut environment is the gut microbiota, where and if there is a disruption in the barrier itself, it may actually cause a direct activation with the rich immune cells, especially the T regulatory cells, as well as the nerve cells. Another important stimuli is the food antigens, and because of these food antigens, it may result in induction of the innate immune response, but it may also have effect on the amylase trypsins, as well as production of other fermentable substances, for example, short-chain fatty acids, which may also have an effect on the enteric nervous system itself. Now, a little break just to promote a textbook for those of you who are interested on everything related to neurogastroenterology and motility. So I was the editor for this book, which we published more than 2 years ago. And there are some very interesting, I would say, chapters which will enhance the knowledge on the gut-brain axis. Now, we heard a lot about gut microbiota, and this is certainly a fascinating new branch of gastroenterology, but it has also opened up to other fields in medicine. In fact, what we know about the gut microbiota, at least in the medical school, is all related to culture. But because of the modern technology of the DNA sequencing, it has made it even more possible for the scientists to understand the function and the role of the gut microbiota. Now, along the GI tract, there are differences in the abundance and also the composition of the gut microbiota, where obviously in the stomach, you can find the Helicobacter pylori, but also a dominance of lactobacilli. But the numbers are clearly smaller because of the presence of acid. And likewise in the duodenum, the amount is also much more smaller and limited to only several types or species of a microbiota. But this microbiota richness tend to increase distally and even more so in the colon, where you can find more abundance, even up to 10 to the power of 10 to 12. Very importantly is this balance in the composition and abundance is the basis for a healthy gut. And this relates to the concept of eubiosis, which is a normal microbiota balance and composition versus a dysbiosis, where there is an imbalance in the gut microbiota composition and the abundance. Interestingly, the gut microbiota ecosystem itself starts even from the birth, and within the first 1,000 days of life, it's really a greatest window for a microbial modulation, and it tends to stabilize at the age of 3 years old. So in healthy adult, usually the composition will be stable unless it is disturbed by antibiotics. But when it comes to elderly, then again, the microbiota will change, resulting in a much more lower diversity in terms of a species composition. And all of these changes throughout life on the gut microbiota abundance and composition, they have a short-term or even long-term health effects. Now, I think there's several myths, when it comes to gut microbiota, and the numerous publications have sort of like helped us understand what are some of the factors that determine the abundance and the compositions. So in this particular paper, which we published by our own working group on the GI health, we actually screen through the literatures as well as to collect data and experiences which relates to the role of this gut microbiota in early life. Very important is that, as I mentioned early on, newborn babies are not sterile, but they have been already colonized even since the conception or the fetus level. And Sicilian section itself, it has been shown to cause an alteration in the gut microbial colonization. Breastfeeding and also infant formula may have a differential impact on the gut microbiota. And the use of antibiotics during pregnancy and infancy have been found to affect the gut microbial colonization. Most importantly is that there are numerous environmental factors, or what we call as the exposomes. For example, farms, pets, even politics, and football matches. All of this can play a role in the modulation of the gut microbiota. And I did bring out the term dysbiosis early on, and it has been shown that when there is an imbalance in the gut microbiota abundance and composition, it may have a short-term impact as well as a long-term impact. Among the short-term impact would include, for example, diarrhea and clostridial difficile associated diseases. But whereas the longer-term impact may include obesity and other metabolic disorders, including diabetes, but also inflammatory bowel disease, asthma, allergy, and so on. So the question is that what actually determines the gut microbiota composition? Is it the diet or is it the genes? So in this particular study where we collaborate with the National University of Singapore, where we administered the food frequency questionnaire and collecting stool for metagenomic analysis from a group of primary school children between 7 to 11 years old, and the males consisted about 54%, with an almost equal representation of two major ethnicities. And we did find that in this particular cohort, obesity was found in almost a quarter of the primary school children, which is quite alarming. Just in case you're not sure who it is, this is me, using the opportunity to give some education to the parents and the children during this particular, field study. So interestingly, what we found was that Kelantan Malays and the Chinese, basically, these are the two ethnics that stay in the same area in Kelantan, and they have similar bacteroides to Prevotella ratio. And when we compare to the Chinese in Kelantan and the Chinese in Guangzhou in China, obviously they are very different. The Chinese in Kelantan, they have more Prevotella than the Chinese in Guangzhou. And likewise, even in Malaysia itself, in Penang is another state on the other side in the west coast of Malaysia, or peninsular Malaysia. It seems that despite the differences in ethnicity, but Malays and the Chinese, they share very similar gut microbiota. And therefore, this study has shown that the enterotype or the gut microbiome of a population is largely dependent on the geography where you stay, and not really due to ethnicity or gene. Basically, food is the most important factor that determines the gut microbiome, and therefore you are what you eat. Of course, it begs the question on what is the most beneficial diet. In fact, we have prepared this review, invited review by Jackie. Jackie is one of the top journal in the area of allergy, where based on available data, we do observe that certain diet may be more beneficial, and this may include a Mediterranean diet, as well as a traditional Asian diet, where all these diets are characterized by really high fiber, but also consists of functional food, which is enriched with probiotics and so on. Now, this type of diet with high fiber and functional food, it helps to promote the growth of beneficial microbiota, especially Prevotella, which is important in the digestion of fibers, and also promote production of very beneficial metabolic byproduct, especially the butyrate, which we all know to have anti-inflammatory effects. So let us go back to the case study, and let's talk about irritable bowel syndrome, or IBS. Now, it has been a myth that in the older days, a physician tend to separate the physical body from the brain until 16th century in Descartes. And this concept of separation of mind somehow has become very dominant and pervasive in the Western thinking. And even until today, many doctors would first of all, really separate between the two, the mind and the body, and we will think everything as being a disease or organic. But we know that very often and organic disorders also often have a functional component, meaning the brain component. And because of this separation, it often has a very profound negative effects on research, but also patient care, and more importantly, the patient-physician relationship. And this is also the basis of functional GI disorders. And from the very beginning, we, as a physician, we should treat a patient as a whole. And that's why this concept of biopsychosocial model, where we incorporate not just the biology or the physical, but we should also have the incorporation of the psychosocial component in order to explain an illness. Now, for 3 decades, the working group have been working on this term, this disorder, the functional GI disorders, where we collect more scientific evidence to prove that both the gut and the brain do not exist separately. Very often, of course, we know now that the functional GI disorders, or what we term as disorders of the gut-brain interaction, are really consists of interaction or a combination of GI motility disturbances, visceral hypersensitivity, changes in the mucosa and immune function, gut microbiota, as well as the central nervous system processing. So very often is also we found that the functional GI disorder, and through also to other chronic pain conditions, they are associated with a reduction in the neuronal density in the brain, pain control areas. Then the psychological treatment that we provide may actually reverse this process and regrow the neurons in what we term as a neurogenesis. And sometimes the treatment may need to be given in combination in what we term as augmentation of medication and psychological treatment in order to sustain the response for even longer period of time. Now, I'd like to illustrate a true story that I personally have encountered, where back in 2014 in December, we had the worst flood in this region of the Peninsular Malaysia where I stayed. This is also the state of Kelantan, which I mentioned early on, and here is Penang. Now, during that flood, more than 200,000 people were affected with broken houses, with no clean water and no electricity. So my family and myself were also a flood victim at that point of time. We were trapped for 1 whole week, and nothing other than water surround us. So it was really a difficult period for many of us. In fact, many fell sick with dengue, typhoid, but also acute gastroenteritis. And in a community study that we conducted, it appears that more than 30% of those flood victims would have developed acute gastroenteritis. So we used this opportunity to carry out a community study about 6 months after the flood in 2 villages in Tumpat. Obviously, Tumpat is where the worst affected areas. And what we have done is to administer surveys and also to collect the stool specimen for metagenomic analysis. And for those flood victims with irritable bowel syndrome, we gave them a probiotic for 3 months. And what happened was that we found about almost 50% of them developed new onset of irritable bowel syndrome. And those who have an irritable bowel syndrome post-flood had a dysbiosis. And these bacteria, like the Pseudomonas and Trabucella, these are environmentally derived because of poor water sanitation and hygiene. And when we prescribe probiotic for 3 months, it does help this group of patients. It does reduce the symptoms and also improve their quality of life, especially the mental health. And the mechanism is likely a restoration of the microbial balance as well as restoration of the gut-brain health. Because of this research, we actually won an award, and this is the photo that we have taken quite a number of years ago during the award presentation. So let's go back to the case study, and finally, we're talking about the neurodegenerative disorders. So these are some of the common neurodegenerative disorder which has a gut-brain connection. These include Parkinson's disease, Alzheimer's, multiple sclerosis, Huntington disease, and autism spectrum disorders. So for today's presentation, I would probably focus more on the Parkinson's disease and the Alzheimer's disease, which is basically also represent over here, right? PD, which is a Parkinson's disease, Huntington disease, and Alzheimer's disease. And basically this diagram illustrates that all of these mechanisms, including inflammation and dysbiosis here, all of this may actually provide the connection on the gut-brain in this group of neurodegenerative disorders. So this table illustrates some of the microbiota changes that are related to the various neurological disorders, including, as I mentioned, the Parkinson over here and the Alzheimer's. So there are, in fact, many evidence pointing to changes in the microbiome in neurodegenerative and neuropsychiatric disorders. And this may even serve as potential biomarkers, but the only problem is that the actual mechanisms are still unknown and lacks, and therefore there's still a lot of work need to be done in this particular area. And because of these changes in the microbiome, there is a role for microbial intervention in the form of probiotics, as well as fecal microbiota transplantation, both of which I will mention a little bit more detail later on. So first of all, let us visit an old friend, the Helicobacter pylori, which has existed for a long time. It's an ancient microbiome and how it relates with Parkinson's disease. Now, we have found that in this particular study, Helicobacter pylori is more prevalent in the Malaysian percent compared to controls in 21%. So essentially, there is about 1.5 to 3 times more infection in the Parkinson's disease. Also, those infected Parkinson's disease would have worse motor functions, and eradicating the Helicobacter pylori itself seems to improve motor function as well as improving levodopa absorption. The mechanisms are probably unclear, but there may be a direct neurogenic inflammatory process, but also there is may potentially a functional changes of the Helicobacter pylori to the gut, and this then result in increase in the alpha-synuclein aggregation, and also the blood-brain barrier impairment, and leading to the inflammation and degeneration in the brain. So how exactly, or what are the evidence that have been so far demonstrated for the involvement of the microbiota gut-brain axis? Now, first of all, is that there are certain dysbiosis feature in Parkinson, and this include a lower level of Prevotella, which you have heard before, is a fiber degrading bacteria, as well as a lower level of butyrate, which is an anti-inflammatory metabolic byproduct of the organisms. Also, it was found that in Parkinson, there's increase in the amyloid light fiber producing microbes, including bacillus and Escherichia. And this bacteria actually produce very similar protein of fibrils that look like the amyloid in the amyloidosis. Now we know that alpha-synuclein is the one that aggregate in the brain causing Parkinson's, and this alpha-synuclein seem to misfold or misaggregate or even phosphorylated even at the level of the gut itself, and this then migrate to the brain using routes including the M cells and the enteroendocrine cells. And more interestingly is that it was found in Parkinson that the alpha-synuclein in the gut and the brain have the ability to sit like a prion. It not just sit on what type of protein, but it can cross sit into many other type of proteins. So there is evidence to suggest that vagus nerve is the main pathological transport route, and in PD, by performing a truncal vacuotomy, it does seem to reduce the risk of developing the disease. Now in Alzheimer's, there's some similarity and some differences with the Parkinson's. The dysbiosis seem to be amount of the butyric producing organism, including the prevotella, and higher levels of amyloid producing organism. There is a significant loss of the cholinergic neurons, not just in the enteric nervous system, but also in the central nervous system. Obviously, there's also this tau protein and the amyloid protein of causing misfolding, phosphorylation, and aggregation in these two central nervous system and enteric nervous system. And again, this protein, they have a prion-like seeding. However, what is interesting is that different tau isoforms seem to be found in both ENS and the CNS, although the differences in the tau isoforms are not entirely clear on the clinical significance, because it does seem that by doing a truncal vacuotomy did not seem to reduce the risk of dementia, unlike what we observe in the Parkinson's disease. Now, with regards to intervention in modulating the microbiota gut-brain axis, it has been shown that with the use of probiotics, in Parkinson's, for example, with the use of multi-strain probiotics here, including the Lactobacillus edistophilus, Bifidobacterium bifidum, and Lactobacillus reuteri, all of this given for a period of 4 to 12 weeks, will result in a better bowel movement in the patient with Parkinson's disease. Whereas in Alzheimer's, when you are giving probiotics, again, all these multi-strain probiotics given for about 3 months, it does help to improve the cognitive function as well as a reduction in the inflammatory markers and improvement in oxidative stress and so on. Now, the next is a fecal microbiota transplantation, which is something a bit more recent, although such a practice may have been available since the ancient time, but only in more recent years that it has resurfaced because of increasingly resistant Clostridial difficile infection. Now, in fecal microbiota transplantation, it's very important that we need to find really donors which can provide the so-called superbugs, where this a group of healthy microbes would then be mixed, blended, filtered before we re-administered them through endoscopy and even more recently is available as FMT oral capsules. The mechanism of FMT may involve multiple modulatory steps, including the microbiome, but also the host bile acid metabolism changes, as well as the modulation of the host immunity. At the moment, the FMT is only approved for resistant or refractory Clostridium difficile, but there are promising studies for the use of FMT in inflammatory bowel disease. Whereas in Parkinson or Alzheimer's, there are actually no human studies, but in animal models, FMT has been shown to improve cognitive deficits, reduce brain deposition of the amyloid protein, reduce in the tau phosphorylation, and also reverse changes in the microbiome. Lastly, I'd like to touch very briefly on the COVID-19 since we are still in the pandemic. So the mechanism of COVID-19 causing gut-brain disorders is probably still very much under investigation or unknown. But possible mechanism would be that the virus itself may have proliferate in the throat, but also able to proliferate in the GI tract because of sharing of similar receptors. In fact, there is abundance of ACE2 receptors in the GI tract itself. This may worsen if there is a use of a proton pump inhibitor, which reduce the acid and therefore reduce killing of the virus. And by sticking of the viruses to the ACE2 receptors in the GI tract, this would then probably associated with the dysbiosis and somehow interact with the enteric nervous system, resulting in diseases state including post-infectious functional GI disorders, inflammatory bowel disease. And it may also have indirect brain effects, either through direct invasion or blood-brain barrier breakdown, or even immune mechanism. And all of this may result in what we term as the COVID-19 associated dysfunction in the gut-brain axis, or to some of us known as the long COVID syndrome. A bit of revisit on COVID-19 very quickly. We know that respiratory manifestations are most common, and the GI and neurological symptoms tend to emerge later. And more recently, with the Delta variant, it seems like the GI and neurological symptoms play a central role. So some of these GI symptoms will include anorexia, nausea, and vomiting, but also diarrhea and pain. For the so-called long COVID-19 neurological disorders, may include a condition like a chronic fatigue syndrome, and sometimes it can also result in more catastrophic disorders like stroke and encephalitis. Now, very important is that some of these GI and neurological symptoms may actually be the first manifestation even before fever or respiratory manifestation, as we recently seen in the Delta variant, but it may also represent that a more severe form of COVID-19 infection. I did mention briefly about the COVID-19 associated functional GI disorders. And this is a form of functional GI disorders, but usually of acute onset and obviously following the COVID-19. There is evidence to suggest that there is laminar propionate infiltration, increase in the fecal calprotectin, increase in inflammatory markers including interleukin-8, and also intestinal dysbiosis for causing the COVID-19 associated functional GI disorders. In fact, when we were looking at the impact of COVID-19 on irritable bowel syndrome, we found that 11.3% self-reported IBS. In about 4.7% are new onset, with 2% of them or half of them having diarrhea. In fact, during this pandemic, 11.8% seem to worsen, although there is also quite quite a number of them did improve the symptoms. And all of this may be related to the more flexi hours, the increase in social well-being, or the less work stress. Whereas for the long COVID-19 with regards to the Parkinson, I did find that in this particular publication published quite recently, which is a series of 23 patients recruited from several centers from the UK, Italy, Romania, Mexico, between March last year to March this year, where almost 85% of the patient with Parkinson's disease develop a long COVID-19 syndrome. And half of this Parkinson's disease, they have worsening motor function and increased levodopa requirement. And other common symptoms include fatigue in 40.7%, cognitive disturbances in 22%, and also GI symptoms. Now, some of these long COVID disorders in Parkinson's are not related to the severity of the COVID-19, but a combination of disturbances in the psychology as well as the COVID-19 related viral syndrome. So with that, my colleagues and friends, I'd like to conclude that the gut-brain axis is really a complex bidirectional communication that is vital for homeostasis and health, and the gut microbiome is becoming an important part of this axis. Understanding the structure, physiology that underlies this axis may help to explain mechanism and treatment of diseases, including the neurodegenerative disorders. I'd like to end to say thank you to all my fellows, my PhD students, my other trainees for their contribution to the research. So what we achieved today, we owe it to our great team members. And lastly, just to promote a meeting, the Asia Pacific Digestive Week, which will be held from 19 to 22 of August. So with that, I thank you very much for your kind attention.
Thank you so very much, Dr. Yang. It was a really, really good presentation from your end. If you're okay, can we just start with the question answer session?
Of course, yes. Thank you very much.
Thank you very much. Thank you. So the first question that we got up till now is, can the recovery of gastrointestinal symptoms help in managing the effects of neurodegenerative diseases like AD or Parkinson's?
Right. So I think the evidence does point that there are certainly connection between the two, and really that the science behind the gut-brain axis and the role of microbiota are very recent, but they are gaining traction and gaining interest among many researchers around the world. So it's always, when a lot of people are doing research on it, and we tend to find association, but whether there is a direct causal relationship will really need more robust, clinical studies. Sorry, I think we couldn't hear you actually.
Okay, so am I audible now? Right.
Yes.
So the next question that we have is, what negative effects are seen on gut and neurodegenerative diseases? Research has shown due to the misconception of mind and body myths.
Yeah, I think this is a fantastic question. I think for a long time, people would have thought that is whatever that happens in whatever the gut symptoms that happen in many of these neurodegenerative disorders are basically what happened in the brain, but really it's probably an effect of both the gut itself and also the brain. It's very interesting that based on recent research on how all these tau proteins and other protein like amyloids and alpha-synuclein, they can be found in the gut itself, and how these proteins are also found in the brain, and then how these proteins are actually connected to the gut microbiome. So I think all these findings are certainly interesting and may provide some explanation on the pathogenesis. Although I would say that whatever study that we have at the moment are very early and certainly merits more research. Yeah, I hope that actually in a way answered the question.
Yeah, it adequately did answer the question. Proceeding with the next question, Devesh Kumar Mishra asks us, can a patient suffering from Alzheimer's have any improvement upon feeding with pre or probiotics?
Yeah, again, a very interesting question, because the use of this probiotic is not something new. All of us, I'm sure, at some point are taking probiotics. In the form of yogurt and lassi and so on. So these are what you call as a functional food. In fact, if you believe in this, if you have been taking it, especially in many parts of Asia, including I'm sure even in Pakistan, in India, even in our part of the world, for those people who have been taking functional food or probiotics, generally it will, of course, preserve our gut health, and also it somehow leads to a less decline in the cognitive function, right? And therefore, maybe it's not surprising that someone who have dementia or even mild cognitive function may actually do better if they are on some form of probiotics. But bear in mind is that but for those who already developed dementia or Alzheimer's itself, if you were to start taking a probiotics, there's actually no study to prove that it will improve the Alzheimer itself. This is partly because all of this will certainly takes time. We cannot take a probiotics, when the disease has already happened, and hopefully it will reverse. I think this is extremely difficult, but if we have been taking it even from the very beginning as a form of preventive medicine, I think that would certainly help, rather than using a probiotic, at the point when the Alzheimer's has already developed. Although I must say that I think we should have more research on this. Although doing a research such as this will take a long time to determine a beneficial outcome.
Okay, it was a really good explanation. Thank you very much. The next question that we have is, can a behavior of autistic individual be controlled by controlling their diet? And if so, what is the mechanism behind it?
Yep, great. And in fact, I think a lot of people are paying attention to autism. We don't exactly know, of course, how is the connection between what we eat and also what it determines the behavior. But I'm sure many of us would find that eating chocolate, make you happy. I mean, I do have many friends who like to take chocolate and it makes them happy. So in a way, the diet itself indirectly really helps to determine your mood or your behavior. To some extent, I think there are compounds in the diet that may really help in modulating the mood and the behavior, including those children or patients with autism. So say, for example, I did mention in my talk, if you have a food that contains more fiber, right, or functional food, both will promote the growth of healthy or beneficial gut microbiota, and this will then produce good byproduct, including butyrate, which has anti-inflammatory properties. And because of that, it does seems to help in reducing allergy. And not surprising also that if you were to take this that kind of diet, it will probably also improve the mood and behavior, including even autism. So, but again, these are early data, and I do think we need, again, more robust data or research to really back up the, the claim.
Right. So the next question is from Joshua Wilson. He's asking, in syndromes such as PTSD, patients cannot eat many necessary foods like milk, carbohydrates. So will this make them vulnerable towards neurodegeneration process?
Well, this I think interesting, and I do agree that not everyone would be able to take a food as what we call as FODMAPs. In fact, many of our diet that we are taking today are enriched with very simple molecules and high in carbohydrates. So these foods often, although will provide short-term energy, but they may not provide all the important nutrients needed for the health of the gut microbiome, or even for the brain health in that sense. Now, of course, when it comes to diet, there's always this a psychological factor that we need to consider, and also availability of the so-called diet. I think someone who stay in the city and someone who stay in the rural and plant their own food will probably have a very different ways or even responses to the diet that they are taking. I'm not sure exactly the interaction between, say, the food that the question is asking and PTSD, but certainly again is very similar to what I've answered with regards to the autism. There is a connection, but you probably need to be proven.
Okay, so now we have two questions. They're certainly similar, one from Joshua Wilson again, another from Asia. They are keen to know, to what extent intestinal permeability affects gut microbiota, and what is the relationship between depression and the permeability of the intestine?
Yep, so excellent question. In fact, the so-called gut barrier, it's the meeting point between the external environment as well as the internal environment. Any disturbance in the gut barrier will cause either a change to the gut microbiome, or maybe the gut microbiome actually also changing the gut barrier. So it's sometimes a chicken and an egg. Both can play an important role in most importantly, is really the balance, a homeostasis connection between the two. And there have been a interesting connection between what happened to the axis, the microbiota gut-brain axis, in causation of depression. Now, if you have read about the history of depression, you realize that at some point, people are treating depression in the past with antibiotics, and they found that some of these depressed people actually cured after taking antibiotics. So now, whether that is a real science or not, it can be challenged. But if you really want to approve whether the modulation of the gut microbiome will treat depression, we will certainly need good clinical trials in that regard.
Okay, one more question that I want to ask you is there a link between the decisions we make from the brain and from the gut? Are they any ways different, or is there a similarity between them?
Oh, excellent. Well, I do think that the gut and the brain, they keep talking to each other all the time, just like the husband and the wife. But although sometimes if if they have a little quarrel at times, so it's not surprising that they may they can get some disconnection or in imbalance. But once they are talking again, I'm sure the gut and the brain will result in a better health and a reduction in disease. So I guess very important is that we need to keep the husband and the wife to keep talking and not create any disharmony between the two. Yes, it's an analogy.
Okay. So can you please specify how microbiota gut product actually interact with the blood-brain barrier and cross it?
In fact, I think I shared also in my talk that the both at the gut and the brain, they actually come from very similar and biological development, and because of that similar development, they share many similar receptors, although they may have some differential effects. When we activate receptors in the gut may have a different effect by stimulating the same receptors in the brain itself. So I think although that they have some similarity, but certainly the gut and the brain also have some differences. So in that sense, I think it will explain why the reasoning behind the question from that you have asked just now.
Okay, thank you very much, Dr. Young. I guess we are ending here the question answer session. It was really humble of you for joining us for the session. Thank you very much.
Thank you very much. And yes, I look forward to, also research collaboration. Thank you. Thank you very much. Bye-bye.
Dr. Nikhat Ahmed Siddiqui. She is a meritorious professor of neuroscience from University of Karachi. She has served University of Karachi for over 33 years and has provided her valuable services in Ziauddin University and Beirut University. As a Dean of Faculty of Sciences and a researcher. She is an experienced Dean of Faculty with a demonstrated history of working in education management industry. Dr. Nikhat is skilled in clinical research, life sciences, strategic planning, thought leadership, proteomics, and genetics. She has obtained her PhD focused in applied neurosciences from a different Department of Biology and Health Sciences, University of Surrey, Guildford. She did her postdoctoral from the School of Medicine, Tufts University, Boston. Dr. Nikhat Ahmed is a founder member of Pakistan Proteomics Society. She is a pioneer in the field of proteomic research in Pakistan and authored over 80 scientific papers in prestigious international scientific journals. She has contributed international projects on proteomic research and has developed regional map of brain proteomes from 80 patients. She serves on the scientific and advisory committee of numerous academic and private research organizations. Dr. Nikhat is already with us, and she'll be conducting a live question-answer session, which here we are delivering a lecture, which she have already recorded. So we'll continue with that.
Assalamu alaikum and a very good morning. Thank you for inviting. It's a pleasure to be here and sharing my thoughts on the future dimension of research in neurodegenerative disorder. I'm going to launch right into a quick introduction of neurodegenerative disorders, and this will carry us all the way up through the current research. So neurodegenerative disorders are basically incurable diseases, and there are no disease-modifying agents and hence no cure, despite an increased economic burden, which accounts for 1% of the total world GDP. You have been listening to a lot about neurodegenerative diseases, and I will talk briefly about the primary brain regions which are affected in major neurodegenerative diseases. What are the major neurodegenerative diseases? Alzheimer's disease, Huntington disease, Parkinson disease, and ALS, amyotrophic lateral sclerosis. These are diverse group of conditions, including most common, leading to neuronal loss and cell death. You can see the brain areas which are involved in different functions, like cerebral cortex involved in executive function is affected in case of frontal temporal dementia, Alzheimer's disease, and Huntington's disease. Similarly, basal ganglia involved in movement and reward in case of these neurodegenerative diseases. Thalamus, this is a sensory gateway involved in frontotemporal, Alzheimer, and Parkinson. Hippocampus, that is memory, call, cognitive function, learning, that is affected in case of Alzheimer's disease. Cerebellum, which is controls the balance and movement, is affected in case of spinocerebellar ataxia. Then basic body functions controlled by brainstem is also affected in ALS and LCK. And all this leads to neuronal cell death and cell loss. If you look closely at the number, at the percentage of population, you can see that this is a source from United Nations World Population. You can see that change in the makeup of the population from 1950 to 2040 or 2050, which is not, I don't think you can see that, but it's true if you believe me, that as the life expectancy of developed countries increases, Alzheimer's disease and Parkinson disease will take over cancer to become the second leading cause of death after cardiovascular disease. So we know that the primary risk factor for neurodegenerative diseases is aging. You can see here the percentage age uh, 65 or over, and this is predicted for 2050. So this is going to be our world in 2050. Yes, of course, this is a much better place compared to in case of this, but there are other type of dementias which are prevalent. So this shows the aging population, and this aging population will contribute to the primary risk factor of neurodegenerative diseases. Neurodegenerative diseases result in the cell death, specifically the neuron. Neurons communicate with each other to perform all the brain function, from movement to learning to memory, thinking, you just name it. Because of brain complexity, brain disorder, you can see a very complex brain. Brain disorder can occur from very small miscommunication between the cell. You can see the very closely connected neurons, network of neurons, and if there is a disruption in any one of it, it is going to affect a number of different other connections, and hence activities across the brain. So due to aging, the brain becomes vulnerable to restore and retrieve information, and this brain ability to restore and retrieve information is diminished because of because of degeneration of neurons. Besides, we can see the common mechanisms which are there in neurodegenerative diseases. Most of it, neuron loss, loss of synapses, and formation of protein clumps. So besides the loss of neuron, degeneration of synapses, you can see here a number of synaptic connections, and here the connections are diminished. So they also degenerate, leading to various neurodegenerative conditions. What is causing neuronal death and why? Because loss of connections lead to loss of proteins function. As you can see, the aggregates of protein disrupting the connectivity and hence leading to neuronal cell death. In Alzheimer's disease, it is the amyloid proteins which are deposited, which accumulates, called amyloid plaques. In Parkinson's disease, it is Lewy bodies, which we can see. From this point onwards, the primary goal of the scientist was to get rid of these protein clumps, or the denatured proteins, or the non-functional proteins which are forming clumps, in order to restore the normal brain function. And why the protein is not folded in its actual conformation is because the active protein is converted into a non-functional protein, and it is denatured due to a number of factors which are involved in it. So we see here that is native protein is active and functional, and it is denatured when it loses its function, and hence loses its function. So this concept lead to the emergence of another important concept of molecular form of protein. That is, if a protein loses its shape at any structural level, it may no longer be functional. Plaques in AD, Lewy bodies in PD are not the only factor which are responsible for neuronal cell death. Rather, the disease pathology is driven by other proteins that form clumps and accumulate because of loss of native structure. So structure plays a key role in protein function. And this is going to be my focus of the talk about. I'm going to talk in detail about AD proteomics. Briefly, we will overview the research trends in some of neurodegenerative diseases, which is driven to a great extent with the emergence of high throughput technologies. And so, if neurodegeneration, we know, is associated with the structural changes, like we saw in histology, the disease proteins. So this concept is utilized in drug development and for better understanding of the mechanism underlying neuronal loss. So if we can identify the disease protein causing progression of disease pathology, it will help in development of effective therapeutic agents. Here, the technological advancement has helped in achieving this goal. So, the dimension of research should be broadened to control the progression of disease. The increasing number of modalities and techniques help provide identifying neurodegenerative diseases at an early stage. And it is very important to identify the disease at an early stages so that the onset of the disease can be delayed and the progression of the disease can be slowed down. And this will help in providing diagnosis, refining clinical trial inclusion criteria, etc. So there are techniques like brain imaging techniques, which is used in identification of biomarkers in neurodegenerative diseases. Techniques like PET, positron emission tomography, MRI, magnetic resonance imaging, These techniques also led to the development of the new dimension of brain connectomics. So now we can see how the different brain regions, how different brain cells in different parts of the brain are interconnected to each other through this connectomics MRI. Then beside this, there are fluid biomarkers in neurodegenerative diseases, and recently we have seen that role of CSF total tau and its diagnostic significance in AD is used to differentiate between primary and secondary tauopathies. Genetic analysis of Alzheimer's disease and Parkinson's disease also pointed out the overexpression of the primary protein deposited and give rise to disease APP duplication causing Alzheimer's disease, SNCA duplication causing Parkinson's disease, and MAPT duplication causes tauopathies. There is one important development in biomarker brain imaging technique, that is biomolecular fluorescence complementation, which determines the aggregate formation in real time, just like real-time PCR. So the scientists have detected TDP43 and FUS aggregates, which are the hallmark protein of ALS. And this is a very good news that in this way we can move on to validation and hence development of new therapeutic targets. Diagnose the stage of disease, identify appropriate people for clinical trials, and show changes 20 years later. So this is one which has been developed for blood biomarker. The research is still on for this, and you know that this is how the world moves. So where do I, as a researcher scientist, come in this story? I have a long-standing interest in studying how thousands of proteins in a neuronal cell can maintain the structure and function, particularly during aging and disease condition. So my interest was developed in protein profiling, is differential expression in health, in Alzheimer's disease, and in healthy brain, and PTMs, that is post-translational modification of the protein, and how PTMs contribute to protein aggregation, that is through phosphorylation, glycosylation, nitrosylation, that changes the properties of the protein from active to inactive protein. There are a few papers which are related to this, which we worked on the phosphoprotein profiling, and we also showed the differential S-nitrosylation of proteins in AD and differential expression in different brain regions of the protein. We looked through KEGG pathways and Panther pathways analysis, the canonical pathways which are perturbed during the abnormal phosphorylation and abnormal S-nitrosylation. Now this is the current research dimension we are approaching, that is characterization of protein complexes. Now this is a very recently published paper in which we have shown the protein complexes from Alzheimer's disease and healthy brain by mass spectrometry based proteome analysis. Now, due to lack of data on protein complexes, their interactions and implications on Alzheimer's disease, we tried to explore the interacting components of protein complexes and their impairment subsequent to expression aberration in Alzheimer's disease. So here you will see the technique which we use was a native page. This is blue native page, and 2D proteome of BN-derived complexes, where the first dimension gel was orthogonally placed on the second dimension to determine the proteins which are present in the complexes. So this identifying separation of protein control isolated from human prefrontal cortex, we move to the determination of canonical pathways. That which pathways of differentially expressed protein are actually affected in case of Alzheimer's disease. So if you work on KEGG and Panther or IP analysis, you can find out through IPA database, the different types of pathways like oxidative phosphorylation, glycolysis, mitochondrial dysfunction. These are altered in Alzheimer's disease. And Fisher exact X was used to calculate the significance of this. We looked up at the overlapping canonical pathway and major functional domains. So impaired carbohydrate metabolism, you can see glycogen, gluconeogenesis, and mitochondrial dysfunction, oxidative stress detoxification, nucleotide metabolism. These are all functional domains which are correlated with these canonical pathways. So this shows the common protein which exists. Then we moved on to the network analysis. As I told you that we were interested in protein complexes, we want to identify the protein complex, then break this complex into its individual protein component so that we can identify the interacting partners at a time. And if there is a disruption in this interaction in any one of the protein, it will lead to a disruption at a critical level. So we did this bioinformatic analysis of ingenuity pathway network, which is associated with cell morphology and function. So we can see the MAPT and APP are involved in the development of neurological disorder which alter the cell morphology in AD cortex. So there are some indirect interactions with APP and MAPT, and there are direct interactions with APP and MAPT. So this network led us to a better concept of how the protein interactions are regulated and how these interactions, disruption in interactions, can lead to subsequent aberration of expression in case. Again, I implicated in cellular signaling and cell death. So here we can see the network implicated in cellular signaling and cell death, that there are a number of proteins which are indirectly interacting and directly interacting, which are involved in signaling cascade pathways and in neuronal loss. So this is the disease. These are the diseases and biofunctions: cell death, the proteins involved, neuronal cell death, abnormal morphology, development of abnormal morphology of the nervous system, and the progressive motor neuropathy. So the proteins we have identified are involved in a number of diseases and a number of biological functions. So merged disease network shows that it is involved, these proteins are involved in Alzheimer's disease and also at the same time involved in Parkinson's disease, and they affect the cell viability, they cause cell death, and in case here you can see they cause degeneration of neurons, which lead to apoptosis of neurons. In case of Alzheimer's disease, the transport of the molecule is affected, which is expected. Then abnormal morphology of the neurons. So the structure of the neurons are also affected. The quantity of the cells is also affected. The number of neurons are affected. Okay, so this gives you a better understanding of the mechanism which underlies the pathology of progression of Alzheimer's disease. Okay, so this is what is important, and I would like to discuss is the pivotal contribution of apparently expressed proteins in various molecular and biological processes, chiefly autophagy, leading to neuronal degeneration in AD. We use autopsy brain samples, and I will discuss about autophagy as the proteins involved in its mechanism. We found some proteins, a number of proteins, but these are the three important proteins which have not been reported previously. That is V-type protein ATPase, ATP6-VOA1, MAP1B, that is microtubule associated protein, HSP90A, that is a chaperone protein, you all know that. And a new mechanistic finding of this study is increased expression of the V-type proton ATPase, which is critical for decreasing the intraluminal pH during autophagic vacuolar clearance for optimal protease activation. Also, due to decrease in MAP1B, which is required for microtubule bundling and efficient transport of autophagosomes. Then HSP90A, another protein which stimulates the inflammatory pathway in Alzheimer's disease, in turn affects amyloid beta and tau pathologies. So we propose here that HSP90A, which acts as a chaperone of HSP70 in chaperone-mediated autophagy, a direct crosstalk links chaperone-mediated autophagy to macroautophagy. This implicates that neurodegenerative diseases associated proteins are involved in diverse role in cellular system like HSP90A, MAP1B, ATP6-VOA1, VPVPI, which govern the process of autophagy. This shows the proteins are not working as an individual, rather there is an integrated mechanism through interacting with, and this is important. So this gives a close look how drugs regulation of neuronal cell homeostasis influences the pathogenesis and progression of Alzheimer's disease. Now, what can change the trajectory of Alzheimer's disease? This I have taken from Alzheimer's Association data file, that there is we can delay the onset. So if there is a treatment which is developed by 2025 that delays the onset of Alzheimer's by just 5 years, then we are expected to develop Alzheimer's 2050, that is 5.7 million, would not take place. As we can see, there is a plateau formation. So what important is to identify the disease protein which are causing, and then targeting all these proteins for the treatment of, or they are used as a modified therapeutic targets. So this is what can be done, and I think in near future we will be able to reach here because of advancements in technology and advancement in attractive development. Although no significant progress has been contributed by me, nonetheless gives hope, still hope, that a number of committed students which are trained and mentored will make societal contribution in future. So this work has been contributed by my PhD student Beena Hassan, Sadia Zahid, Aisha Khan, and by Professor from Department of Biochemistry, University of Karachi, Professor Dr. Asif from Institute of Clinical Chemistry, Göttingen, Dr. Christoph Lenz, Institute of Clinical Chemistry, Max Planck Institute, Germany, and they contributed in mass spectrometric using nano LC-MS. We are thankful to HEC Pakistan and for providing the grant, MRC certificate back, University of Edinburgh, UK. Thank you very much.
Thank you so much, Dr. Negat, for such a productive session. Can you please put your mic on the unmute session and come live for us to maintain the question answer? Hello, Dr. Negat. How are you?
Hi, fine. Good morning. Good evening. Good afternoon, rather.
Good afternoon. So, if you're okay, can we start the question answer session?
Yes, sure. Yes, sure.
Yeah. So the first question that we have is, can the formation of protein clumps be delayed? And if so, what is the mechanism behind it?
Well, this is the question all the scientists are looking for, but I would say that there are a number of drugs or therapeutic agents which have been identified and which have been in clinical trials and in use. But the only thing is that it is the protein clump, if we are that, like I said in my talk, that if we are identifying one protein, that is, for example, tau, and if you want to get rid of the clumps of the tau in tau paths, then what is the problem is that there are other proteins which are also forming the clumps. This is the major, what I say, a dimension in AD, that nowadays we have developed a number of therapeutic agents, but so far we haven't achieved any progress in, or any modifying agent in the treatment, or which can delay the progression of disease. So there are some drugs which are being in use now, but I don't think they are so effective in that. But in near future, I think with this technological advancement, we may be able to come up with something. Can't hear you, Minal.
Okay, so the next question that we have is, how can we delay the conversion of active protein into denatured protein in neurodegenerative diseases like AD and Parkinson's?
Conversion of active, this is again the same thing. When a protein becomes, when the structural changes occur, when the morphology is affected, then the protein shape is changed. And like I said, that there is a very firm relationship between structure and function. So if there is any change in the structure at any level, the structural level, right? You know, there are primary, secondary, tertiary, quaternary structure. So at any level, then this will affect the functioning of the brain, and that makes a protein causes aberrant functioning of the protein, and hence the protein cannot properly perform that specific function. So this is active to there are no therapeutic agents so far to say revive the functioning of the protein once they are teenagered.
Okay. So, Dr. Negat, what do you think? Is there a progression or succession in the research related to the management of protein complexes and pathways and their effects on neurodegenerative diseases?
I think a lot of work has been done by a number of people who are working on Alzheimer's disease, and of course coming up with this, looking up at this. But what we did was that since there was no data available on protein complexes in Alzheimer's disease, so we thought maybe if we will go into the details of the protein complexes and how they are altered and why there is aberrant expression and how these complexes, that is what is a complex? That is a number of protein components which are working in concerted effort, right? So if there is what I should say, increase or decrease in expression or differential expression of protein in this case, then what will happen? That one of the protein, if its expression is affected, it can affect the whole functioning of the whole protein complex. Are you getting my point? And it's not, one protein is not affecting one pathway. They are connected, as I show you through the bioinformatic analysis, there are a number of pathways which are involved. So it is going to be really difficult to find out, because it is like finding a needle in the haystack, you know, that you have to find out. But I think there are a number of research which have come to this point where they can, especially with brain imaging, because if we can develop fluid biomarkers like CSF or blood biomarkers, and if we can validate, which people are validating through imaging techniques, then maybe we can come up with something if we can diagnose these diseases at an early stage. The only important thing is the diagnosis at an early stage, because the symptoms appears after 20 years, right? So there is nothing one can do. So if one can identify this, right, if it is say like running in the family or things, those genetic diseases, then if we can identify this at an early stage, the problem, the changes which are taking place, then maybe one can regulate the disease progression. I hope it has answered the question.
Yeah, it does. So do you think there is a role of protein complexes and their association with the seizures?
With seizures, I am not an expert on this, but I've got I don't think I can answer this question because I don't know if it is going to affect. I haven't read anything related to this. Maybe.
Okay, not a big deal. Mr. Shah Heather is asking, what are the major factors involved in protein complex denaturation in AD?
Major factors involved.
Yes, the major factors.
Okay, there are again there are. Sorry.
No, you can continue.
There are the protein complexes are like, you know, that protein working in harmony, right? So if we say that there is an effect, if a person with Alzheimer's disease also has diabetes, for example, right? I'm just giving an example. Then in that case, of course, we know that the carbohydrate metabolism is affected, right? So say if in a complex, we have a number of proteins and some of the proteins are regulating the carbohydrate pathways, right? Okay. Like I told you, like glycolysis, gluconeogenesis, and all this. So if this is what is affected, then of course, this will cause a problem with this can be one of the factors in this. All right. So these comorbidities and things can also be one of the factors, but the protein complexes themselves we are still looking at. that what is causing the change in the protein complex. Because with protein complex, the problem is you have to work at the membrane level. And identifying the protein complex, separating the protein complex, it took us 3 years to get to this result just by working on the bench, right? Maybe people can do it, but we did, we have put a real effort to get to this point, to get the protein complex and then find the protein components. So there can be a number of factors, one of which can be post-translational modification of the protein, like increased phosphorylation, or what we call hyperphosphorylation, or glycosylation and nitrosylation. And we have found that some of the proteins in these protein complexes are both phosphorylated and nitrosylated too. So this has not been reported before. So maybe phosphorylation and nitrosylation is affecting the working of the protein and hence of the complex.
Okay, thank you very much, Dr. Negat. It was so considerate of you to sort your time out for us and for all this conference and everything. Thank you very much for your time.
It was a real pleasure. Thank you, everyone.
Take care. So ending the session now, we'll just take a break for an hour and we'll continue with the lab tutorial after that. So you guys can have a break for now.
Hmm.
Okay, so we are back now and we'll be starting the session soon. I hope everybody's there with us. So we have Dr. Peer Asad with us right now, and he will be conducting a lab session for us. Dr. Peer Asad is an MBBS doctor from Lakat University of Medical and Health Sciences, Hyderabad. He is known for conducting the first ever brain surgery for the removal of acute subdural hematoma with extradural hematoma under the local anesthesia in patient presented with ventricular septal defect fitness and in the critical state. Much of his research work is in the discipline of neurology and neuroradiology and neurosurgery. He is an expert in the field of neurosciences and has been serving for health and well-being for years. Dr. Peer has published several research articles mainly focusing on the isolated intracranial residuum disease and acute subdural hematoma with extradural hematoma evacuation among the ventricular septal defect patients, etc. So he will be conducting the lab session for you, and you guys will attain the knowledge with regards to the neurosciences and untangled neurodegeneration. Over to you, Dr. Asad.
Thank you. So is the screen visible to everybody? Can you hear me?
Yeah, I guess everybody can hear you. You can continue.
So basically, as you know, I'm Peer Asad Aziz. I'm from Hyderabad. I'm working as a senior registrar in the Department of Neurosurgery, Liaquat University Hospital. And so as this session is for tutorial, but before going to tutorial, already there are so many in this session, so many things are already discussed regarding neuroscience. So sorry. So our topic would be the role of neurosurgery and its impact in neurodegenerative diseases. So as I belong to neurosurgery department, and we are the last resort for any neurodegenerative disease to counter. So usually these such kind of diseases are already being discussed and seen by our fellow colleagues like neurology, and other works are being done by basic science departments. So what this is the our city Hyderabad, and in this is a basically the market area and the fort which represents our city. In there the monument of our legend, and he fought for the war of independence. And there is a Shahi Bazaar in the last, which is the biggest bazaar of Asia. And so coming back to the topic of neurodegenerative disease, so we will be discussing Parkinson, Huntington, ALS, Alzheimer's, NPH, multiple sclerosis. All these diseases are being discussed so far in our sessions. So in particularly regarding neurosurgery, what we will be discussing is basically I will put an impact on Parkinson and normal pressure hydrocephalus. Basically, when we are dealing the neurodegenerative diseases, these all Alzheimer's, Parkinson's, normal pressure hydrocephalus comes in differential of each other. They all represent nearly the same to each other, and there are small changes, so that clinically when we see the patients, so these patients usually resemble each other. And before going to into this, first of all, I'll just quickly summarize the basal ganglia anatomy, which everybody should be already knowing, that it is a collection of nuclei which are interconnected, and the importance is in coordinating movements and cognitive functions, and it works with pyramidal motor pathway. So the other components which are corpus striatum, in which we include striatum and pallidum, substantia nigra, and subthalamic nuclei, amygdala, and colostrum. These are the units which usually join together, and then the works which our body represents happen. So the functions are motor cognitive functions, controlling the eye movements, role in motivation, decision making, and working the memory. So where is the clinical significance of these nuclei? And if they do not work properly, there could be hyperkinetic or hypokinesia, and then they become mainly motor and movement disorders and Parkinson's disease, and there are other diseases as well. But as our focus is on Parkinson mainly, and the role of the surgery, what we have seen is very beautiful in such diseases. And basically that is not the actual course is the surgery. Neurosurgery basically is not actual treatment, cure of this disease, but it masks the symptoms. So coming to the point, one feature that we have, which happens commonly and we encounter in our clinics, mostly the neurology fellows used to see these, and then after neurology consultation they come to us, and when they come to us after neurology consultation, we decide that we either these patients need a surgical care, or again they should be treated on the basis of medical therapy. So one of these is dystonia, which are involuntary contractions of the opposite muscles, and the patients usually had such kind of positions that could be cervical or truncal, and the patient's movement and the patient's movement and posture is very much disturbed in this, and along with it, patients had dyskinesias, bradykinesias with it as well. So coming to the historic background, the ancient artwork defines that it was already present in that era in past. And first of all, in 1641, German surgeon Meniscus cut the muscles in the neck for cervical dystonia. So the aim was that around 400-500 years back, what these surgeons were doing, they were considering the dystonia that it is because of muscles. So they used to cut the muscles, and they were thinking that actually this is happening and we are treating that. But at that time, definitely it was not proven, and it was not well known to the community that why these things are happening and where is the problem. So the first applied term dystonia musculorum deformis was described by Herman in 1911. Prior to that, many physicians mistakenly believed dystonia was purely psychiatric disorder. So in dystonia, there are pure primary dystonias or secondary dystonias. In dystonias, primary dystonias, usually what are the issues are dystonia 1 and dystonia 6, DYT1, DYT6 genes that are responsible. And in 30% DYT1, 30% penetrance is there. In initial one limb usually it occurs in one, and afterwards the other side of the body also involved. It responds to the surgery. The surgery, main aspect of the surgery, which these days is happening in the current era, are either the lesioning, which we call it pallidotomies, thalamotomies, or deep brain stimulation. So deep brain stimulation is working much more better, and we will see afterwards in the later part. While the DYT6, there are 60% penetration, and initially it occurs in cranial cervical region. Initially it responds to DBS, but later deep brain stimulation, it responds to deep brain stimulation, means surgery. But later afterwards, what happens? The symptoms again reappears. So how these dystonias are being treated? If we see every kind of disease, when a disease presents, usually initially we deal with conservative and supportive therapies. In conservative, we go for a medical therapy. So the medical therapy used for it is pharmacological things that are levodopa and anticholinergic drugs. And then baclofen is also used. Baclofen is used orally as well as its pumps are used, and which is minimal invasive kind of thing, and with the pump is used to treat the spasticity that happens because of it. And past botulinum toxin and botulinum toxin was also been given, but these are less familiar these days. Although if we see that the role of neurosurgery, and deep brain stimulation in it particularly, or thalamotomies and pallidotomies, lesioning. These are very expensive procedures. So, and machinery which is required is not available in every setup, especially in a country like ours, which is low middle income. So not in every setup these things are available. So wherever these are not available, we prefer either to refer the patient looking at the symptomatology, or if the symptoms are a bit less severe, then we initially prescribe these medications. Usually neurologists do that, and in that, if they require, we can apply baclofen pumps, etc. And then, as we mentioned, that the treatment, surgical treatment mainly, our focus would be on these disease, Parkinson's and dystonias. So the central kind of the treatment, surgical therapy appear, that is pallidotomy. Pallidotomy, in this pallidotomy, what we do is basically we put a electrode, and afterward we generate a current that produces heat as well. So the pallidum, globus pallidum, the section of the globus pallidum, in easy words, if we say that, it gets fired, so that after firing it, the effects which we require are observed. The other is deep brain stimulation, which we will be discussing much more in detail afterwards. In deep brain stimulation, what we do is we put a battery inside the brain. We put an electrode that is connected through a wire, and then to a battery. Battery is placed in the chest, and with this the stimulation occurs. And then if the patient had the problems, then the battery itself works. Otherwise the battery gets off. So in this mechanism it works. So what is the DBS? It is surgical treatment in which an implant, or that what I was calling battery, a medical device is applied, and is called, which is also called as brain pacemaker. It stimulates a specific area of brain via electrical impulses. So in this figure, we can see that, if you see that on the left side, a battery is placed in the region, and then it is thoroughly it is connected then back towards through wire. And in the chest, if you see where 1 is written, the battery is placed, that is called implant stimulator. And then when our body doesn't respond, the signals through electrodes and this wire comes to the battery, it sends signals back, and then what happens, the stimulation occurs, stimulation of that particular area occur, and the symptoms usually mask. This is why we called, and we described in the detail initially, that these are basically not the exact cure of the problem, but it is somewhat, it masks the effects of these symptoms. So it is not the actual cure. So what are the indications to DBS, perform DBS? Parkinson's, essential tremors, dystonia, epilepsy, Tourette syndrome, and obsessive compulsive disorders, depression, chronic pain. These days, it's highly in the centers, like in the modern world, it is present everywhere, but in our country, it is present in some areas. So so everything is not possible, but our surgeons, what they are working on it, are mostly they are working on Parkinson's, essential tremors, dystonias. Recently, a few surgeons have also treated OCDs and for chronic pain as well, but not for every. In epilepsy, the work is being in the process, but not in our country. It is not happened yet that the patients are treated for epilepsy with EVS. So what is the mechanism? Basically, mechanism are divided into 4 categories. Number 1, inhibition of the target. If the target is producing abnormal signals, it inhibits. Or activation of target. Sometimes there is combined inhibition and activation of target. Disruption of pathologic oscillations to restore the rhythmic activity and synchronize these. Usually it involves axonal and neuronal transport. By this, there is decoupling that takes place in the cortico-spinopallidothalamic tracts, and with this, it affects. So again, in this, it is represented that it is GABA-mediated inhibition of globus pallidus internus, and then to treat informational lesion, whatever was the lesion. So components. So now in our part, we are basically focusing the DBS, as I already mentioned, surgical part. So for DBS, what we had electrode, then we had extension, means the wire, and the neurostimulator or a battery. So these are the components, as you can see in the figure. So what are the advantages? So advantages, it's reversible. If the symptoms are there, it can be reversed, it can be adjusted, and it improves the quality of life. See, it improves the quality of life in so beautiful fashion that everybody, every patient with severity will definitely would love to go for it, because it masks so much the symptoms. It helps reduce medication, and it is a relatively safe procedure. In past, when the DBS was started in 1980s and 90s in the modern world, and at that time, and comparison to these days, it's so safe. We had neuronavigation systems, MRIs, CTs, intraoperative, with the help of which we pinpoint and localize the nuclei where we have to work, and there is very less margin of error. But although errors happen during surgery, and due to that there are also problems as well, complications as well, it also improves energy level. So what could be disadvantages? Like any surgery, if we go for a surgery, so what happens when you are implanting a foreign body? Our body reacts to it, and when our body reacts to it, it has a chances of increased risk of infection. And the other disadvantage, which in our country is more than infection, I believe, because infection in the third world countries and low income countries is much more higher in rate than the countries like in Europe and America. But again, the expensiveness of the treatment affects more than the infection. A procedure of a DBS with a battery usually costs around 20 to 30 lakh Pakistani rupees, and the whole procedure usually costs around a patient to around 40 to 50 lakhs Pakistani rupees, or 4.5 million rupees. So it affects too much to a country like us. Although in some areas government is helping, and a few cases have been done with the help of the and support of the government, free of cost. But again, the main issue in this surgery is this implant. The implant is very expensive. Along with it, the machinery which is used is also very expensive. So the whole cost of the surgery is too much, and these are the main disadvantages of the this procedure. The limitations to the surgery are less, but those like the patients who have underwent already ablative surgery. The ablative surgery means lesioning, which we were already discussing, like pallidotomy, in which the stereotactically the lesion is created. Such kind of patients will not get benefit. Or those patients who are pregnant in less than 18 or above 75 years, these are limited for the procedure. So target in dystonias usually is globus pallidus internus. Subthalamic nuclei is less commonly targeted in the dystonia. So if we see this MRI, so the cuts in MRI and then anatomic sections diagrams, what we do is we initially target 2 points. One, if you see, if we can see this, my arrow, I am pointing towards. This is the anterior commissure in figure A, and then towards the posterior commissure. So we create imaginary line, and with this anterior and posterior commissure line, we go halfway in the mid of this. We target, because the globus pallidus internus is a bit far and as you see, that's 22 mm. So what we do is, in the midline of anterior posterior commissure, we can go around 2.5 centimeter, 2.22 millimeter or 2.5 centimeter laterally, and posterior laterally we target this lesion, or target or lesion it, so that the effect which we want should be observed. In the other image, you can see the different basal ganglia region, putamen, globus pallidus externus, and interna. So how this occurs is, initially we do a CT and MRI of the patient. Then in the neuronavigation machine, what we do, the image is being transmitted over there in the machine. So after image is there, we apply a frame. If we see the last image in which is 3D reconstructed, there you can see that the frame is already applied, and two pointers like two straw are being applied. These are means electrodes, which are being needles and probes which are inserted, through which electrodes are being applied to target it. So we target it through it, we pass probes, and then if we see in the first one figure, in the MRI, the exact point is being localized, and it's intra-operative MRI and CTs are taken to exactly localize a point, and then after this, it is mixed with the original, means initial image, to confirm that either we are doing it properly or not. This is called planning of this procedure, and afterwards we exactly get the measurements, how much distance it is and how much laterally or medially, to minimize the error. So technique is, the procedure is used only for patients whose symptoms cannot be adequately controlled with medication. We do surgical implantation, and then targeted area is located by CT or MRI imaging, as we mentioned. Quadripolar leads are connected to stimulator and battery. If you remember the initial image in which the battery was connected through external wire. So once the electrodes are being implanted, it's top wires that run side from the head down to the collarbone, but it is usually in the subcutaneous layer, not outside the body. We pass a passer and subcutaneously, and this goes, and near clavicle the battery is placed. Battery is operated by a stimulator that are implanted, and from stimulator, electric impulses are continuously delivered over wire to the wire and electrodes in brain. Reprogramming should be done in 3 to 4 months. What happens that when this system is working, the patient gets improved. Then after some time, the effects and symptoms are a bit decreased. So you have to reprogram it. And with the battery, which is provided by the vendors, they describe you all that how can you program it. So it's easy. So this is the first, sorry. So this is the first presentation. Actually, I am working in Liaquat University Hospital, Hyderabad. Here we do not perform these surgeries, but in Pakistan there are centers where these surgeries are happening. So these videos which I have taken are taken from the internet sources. So in this, you can see this is the preoperative video, and patient had dyskinesias as well as patient had dystonia, and as we will see further, this patient will move. It will be much more evident that the patient had a bit truncal dystonia along with the cervical portion. And look at this, the patient is in severe disease. He cannot walk properly and he cannot stand properly even. So after 1 week post-operative periods, so how much results? Initially the feet and hands were trembling, but now if you can see, the tremors are improved, The patient who was unable to walk is walking. The truncal dystonias are nearly vanished, and this is the same patient after weight gain in 6 months post-op period. The patient had improved a lot. Doesn't seem that the patient had any kind of issue in past. So these patients improve such dramatically, and the effects are so good if treated properly. And the symptoms, if the patients present with the symptoms mild to moderate, moderate to severe symptoms, then the patients are treated with surgery. Otherwise, initially these patients are treated with medical therapy. So in this was a video of another patient in which so this is another patient, and you can see the patient had cervical dystonia along with abnormal movements. So this is very disabling and very difficult to counter such things. If we see, we can feel that the misery the patients are undergoing, those patients who must be undergoing such issues. So he's preoperatively image. This is the preoperative video, and before implant, you can see that the patient had tremors, patient had dystonia, and with dystonia there's rigidity as well. So it will be difficult for him as well to walk, and the parts of the body are not coordinating properly. So we will see it that in the post-op period how the patient will improve. If we'll check that he cannot use his hand properly, his neck, or his gait is also not good. But this is the same patient after surgery. So what is happening? He has improved much. Here tremors in neck as well. The titubations are there along with it, but his gait, his arm swing has returned. Dystonias have been a bit decreased. So the patient had improved overall. So the DBS for secondary dystonia, like tardive, 68% shows 6 months, on 6 months shows improvement, that is 68%. And cerebral palsy is also being treated through DBS, but the trials are not so convincing, and 24% of patients improve. So to conclude, for dystonia, globus pallidus internus, DBS can be effective therapy for dystonia. Generalized and regional dystonias may have similar outcomes, and Divide 1 shows better outcomes than Divide 6 in the primary dystonias. Some secondary dystonias have good DBS responses as well, and mechanism of disease and therapy are increasingly understood. So coming towards the Parkinson, and in Parkinson there are collection of symptoms, as we see that. So this is being a symptomatology how patients presents to the clinics. So in this video you will see that the patients had gait of the patients, tremors, and the patient's face, how it is, and then the abnormal movements with it. if you see this, how disease is this disease is countering the daily life of the patient. The patient is unable to hold the newspaper exactly, or drink a cup of tea. So, or means they do not perform fine activities like buttoning. They are unable to perform properly because of this disease. And afterwards, the diseases this disease is also treated initially with medical therapy, and if the medical therapy is refractory, we perform DBS and lesioning. In this video, we are at present looking that the patient does not perform fine movements. He is not able to write or not able to draw. And with this, if you see that this lady, she had look at her hand, she had abnormal movements in her hand. So these all features presence in the Parkinson. And the gait. Look at the face of the patient. Patient is mask face, and he had tremors at rest. So these have a stupor. They are stupered, and do not stand properly. Take small steps. The hand of patient, you can see that it is not properly placed, and the steps which the patient takes are more, and no arm swing is there. So these all are symptoms of the disease. So how the Parkinson disease presents in the early stages. So if you see that with this female had reduced right arm swing and step length, you check the step length. We'll see afterwards how it it increases. Then the other patient decrease left arm swing and reduce axial rotation. So the patient do not rotate properly. In another patient, if we see that slower gait, speeds, postural changes, decrease axial rotation and arm swing, these all are being checked. Then we notify this on a paper in our performance. Then if the symptoms increases, the patient had bilateral arm swing has been reduced, patient's step length is decreased, bradykinesia is there, patient will shuffle during taking step, patient's posture is stooped, and patient had increased cadence. In the advanced stages, what happens is the features become more severe. Steps severity, the length of step is much more reduced. Patient had more difficult movements. Bradykinesia increases. Patient had reduced postural control. They tend to fall back, and the stability of the patient is decreased. There's when the patient tries to turn, which is much more decreased, and he had defragmentation. So in another patient, if we see that the gait is freezed and he cannot turn, very beautifully, and this can be seen that patient is unable to move properly. And when you when we give these patients complex tasks, so patients are unable to perform these tasks. In another patient, if we see there are several postural changes and postural control, that is causing reduced walking capacity. Again, the gait, freezing gait during initiation. So how do we treat it? Before treating, again, the patients are treated medically through carbidopa, levodopa, and other drugs. If the patients are refractory, we take the patient, choose the patients depending on the criteria, if he is suitable candidate or not. If he is suitable, we prefer the patient for DBS, deep brain stimulation. So what are the criteria in which how the patient is getting selected? The Parkinson symptom usually last less than 5 years. Patient had on and off fluctuation of dyskinesias. patient initially had shown good response to medication, and the symptoms definitely are severe to continue daily life. So again, the picture shows the components of the DBS, in which there's electrode, extension, and neuro stimulator, which we discussed already. So what are the target sites and effects? So what we do is in the subthalamic nuclei, if we target the tremors, dyskinesia, rigidity, bradykinesia, and gait disturbance get improved. If we will target the globus pallidus interna, again the tremors, dyskinesia, rigidity, bradykinesia, and gait disturbance get improved with both of these. But with thalamus, it decreases tremors, but not Parkinson's disease completely. Only for tremors, thalamus is being targeted. So in this particular image, we can see that the ventral medial nucleus of thalamus is being targeted in the left side, and this is for the essential tremors. For Parkinson's disease, subthalamic nuclei has been targeted, and for Parkinson, in the last image on the right side, and dystonia, globus pallidus internus is being targeted. So how targeting is being done, as I already mentioned, there are two types of targeting, direct or indirect. Indirect, we directly target the globus pallidus internus, for example, in this image. And again, the target is being set from the anterior posterior commission line. The line is imaginary, and then lateral to it, posterior laterally, around 22 to 25 millimeter. Usually target has been selected, and then either lesioning is being done, or the lesioning is done, or the deep brain stimulation is done. So target 1/3 distance forward and posterior end of the pallidocapsular border, and 3 to 4 mm lateral along the line tangent to the pallidocapsular border. So indirect targeting. So how do we indirectly target? Again, the line we select as anterior commission and posterior commission line. From there, in the midline, we take a point, and with this point, we indirectly do this. So what are the indications of DBS where we can get benefit? In Parkinson's, essential tremors, dystonia, epilepsy, Turner syndrome, OCDs, and chronic pain and depression. This is the same slide which we previously have seen in dystonias, like the limitations of the procedure are same if the patient underwent ablative therapy. So those patients who underwent ablative therapy, they will not get benefit, and the patient, if pregnant or in the extremes of ages. Technique is same here as well. The procedure, in the same procedure, the patient is initially refractory to medication, but initially got response to the medication. Then the surgical target is a surgical target set, and the implant is placed, and through CT or MRI we fix it. Plane, as we mentioned, then the planing should be done according to CT and MRI. And in the CT and MRI, afterwards we'll place the lead. Once the planing is done, we put an electrode, and in electrode we put a probe. Then in probe we put an electrode, and then we give the voltages, current. With it, we check the symptoms. We check the symptoms, how it's improving. If the patients improve, we usually give the upper limit as well, that till how much the symptoms get improved. Then in between that range, usually the procedure is selected. Then the range should be in this in between. Then after placing the stimulator, the reprogramming should be done in 3 to 4 months. So these are the same pictures, but in on the right side you can see that the target has been selected in the MRI, and then the frame is applied. So the electrode which we apply is getting the microelectrode recordings, and there are different recordings. So before doing a procedure, a complete team usually is being involved in the procedure, in which it includes a neurologist, neurophysiologist, a neurosurgeon, anesthetist, and psychiatrists are also involved sometimes. And afterwards, the electrophysiologic readings are being recorded. And as we see in the lower image, the readings are different. With these, it produces a bit sound, different sounds. And these readings are in the different, according to different depth, how much deep we go, the readings get changed. And here it is already mentioned in the image. You can see that with depth it increases. So this is a video of so now you can see that the tremors are improved. Patient who was unable to hold the bottle is holding the bottle well. Again, tremors are appearing. So you see that when initially, operatively, the patients are usually awake. Patients are initially, as our brain is usually insensitive to pain. So what we do is we put the frame, and for the placing the frame, we put hooks or screws. For this, we apply locally, inject the locally anesthesia drugs, local anesthetic drugs, and then the frame is applied. And afterwards, taking the intraoperatively planning and placing the electrodes, and then after getting the recording, when we are sure that we are on the right point, we actually do the procedure. But the whole procedure in initial, when we apply local anesthesia, the pain is usually patient get pain free. Afterwards, usually patient is awake throughout the procedure, but in some times we also do sleep awake procedures. Means initially the patient is sleeping through inducing the anesthetic drugs, propofol and other prosthetics like drugs. So after getting these drugs, when we reach the point in the brain, then what we do, we recover the patient back from the anesthesia. So these are the two techniques. Sometimes the patients are being anesthetized completely under GA, or sometimes being locally, the procedure is being done. When the procedure was being done, we saw that the patient had tremors, and those tremors were so much that the patient cannot hold the bottle, and if he tries to drink, he was unable to. Then afterwards, the implant was already placed. So what happened? They started the implant battery to give signals back. What happened? The tremors which the patient had slightly improved, and the patient's tremors were gone. So patient was able to hold the bottle, and he was like drinking it. Afterwards, they checked the maximum limit, and again they discontinued it to cross check it, and again tremors came back. So the battery works like this. If the patient gets tremors, the battery will work. If the patient had no symptom, battery will stop itself automatically. So what are advantages of DBS in Parkinson's disease? As we mentioned, it is not a cure, but it improves the symptomatology. It is treatment that helps relieve motor symptoms and sometimes non-motor symptoms as well. Bilateral DBS is often required to improve gait, although unilateral DBS has marked effect on walking. It smooths out on and off fluctuations. It improves tremors, rigidity, bradykinesia, dyskinesia, and decreased medication. One thing more, that when we are doing DBS, usually single side is performed. Usually because we do not interfere contralateral side in order to save it, to prevent any complication. But there are centers they do both at times. So if we see that the patient had before had dyskinesias, had essential tremors, and afterwards the tremors were improved. So this is the basically procedure as we see. If I'll go back and show you. Okay, this is the stereotactic frame, and this the electrode is being passed. So after getting this treated, the patient had improved in posture and he doesn't tend to fall back. So you saw that the patient had initially, when the patient was being seen, the patient had tremors, patient gait was not that good. Afterwards, patient's symptoms were improved. So what are the side effects and risks with this kind of surgery? So patient gets headache, nausea, vomiting, pain, inflammation, swelling at surgical site, confused, sometimes numbness, speech problems are there if you cannot target the proper nuclei. There could be mood changes, hematomas, ICH, or intraparenchymal bleedings, and movement disorders can again cannot, if the patients did not improve the movement disorders. So what are complications? The complications could be related to hardware like lead migration, lead the wire migration, erosion, malfunctioning, or it gets fractured. The patients get seizures and malfunctioning of neurostimulator. Patients get hemorrhage and patients develop dystonias. What is ongoing these days? These days, the fetal tissue grafts are being used for Parkinson's disease, and it has seen that there are good results being observed, that it is beneficial. A bit in past, adrenal tissue transplantation was also done for Parkinson's disease, but the results were not satisfactory. That satisfactory. Fetal tissue grafts are being done for the same disease as Parkinson, and then Parkinson and Huntington disease, and the efficacy of this graft is good. So the ongoing trials and research are going. So to exactly identify how much improvement a patient gets for this. So coming towards the final part of this our today's session, and in this the disease which we see is normal pressure hydrocephalus. Basically, Alzheimer's, Parkinson's, and normal pressure hydrocephalus patients really present same, nearly same. They all have cognitive decline, gait difficulties, urinary incontinence, And if we see that as the patient degenerative changes occur in old age, so there is compensatory ventricular dilation, a bit pressure is increased. So these are the symptoms usually happening in the normal pressure hydrocephalus. And this was described first by a neurosurgeon named Hakim, and in 1964. So before going further, we will see the CSF dynamics. In normal patient, its CSF production is 0.4 ml per minute, while in NPH it is much more reduced and it is 0.25. CSF volume at the end of day is 150 ml. In NPH it is 300 ml, a bit more CSF volume, but exactly it is not the increased CSF production, it's compensatory. So turnover is a bit more. So what features a patient presents with normal pressure hydrocephalus? In this problem, usually patients presents with memory problems, urinary incontinence, and then the patient had gait problems. So regarding dementias, memory problems, they are usually due to subcortical frontal dysexecutive syndromes, and patients can have memory loss or decreased attention, impaired planning and slowness of thoughts and empathy. Patient get apraxias, aphasias, along with it patient had asymmetric tremors and rigidity as well. Patient develop urinary incontinence, the frequency, urgency, and the patient had urgent incontinence. Because of urinary incontinence, patients are sometimes being wrongly, we can say that patients are wrongly being consulted and wrongly diagnosed, like a patient had the age is there, with increasing age in males, benign prostatic hyperplasia occurs, so it can be confused with it. Patients can have autonomic dysregulation. Patients usually may take diuretics and anticholinergic drugs, so these symptoms can happen with these as well. So how patients gait is in the normal pressure hydrocephalus, this is called magnetic gait. The patient's steps are very small, it gets like sticked to the floor. You can see this in this video, that this gentleman is walking, is taking small steps, and he's not properly taking the lower limb up, and turning of the patient is also decreased. Anticholinergic drugs, so these symptoms can happen with these as well. So what's happening? The patient is unable to lift the leg. He's have severe difficulty in walking. So initially the patients when present, usually the symptoms are like this again: memory loss or decreased memory, incontinence, and gait. The first symptom is usually incontinence. So slowness of gait is responsive to shunt. The first symptom when the patients usually come are urinary problems, and the symptom which improves more initially is gait first, and the gait is improved first, while the memory is improved in the last, or it is less responsive as well. 40% of patients had appendicular tremors do not respond to shunt, and the symptoms are usually in 90% of patients. Initially, patients have unsteadiness, frequently falls, difficulty in turning, initiating magnetic gait, and broad base and short steps. The arm swing is bit maintained, patient hair tone is increased, and patients get a bit rigid. The reflexes are exaggerated, and their weakness is unusual. So if we'll compare the few common problems which we encounter in our clinics daily are number one, NPH, and regarding the old neurodegenerative Alzheimer's diseases, and then following it we will also see Parkinson compared with Parkinson as well. So what happened in Alzheimer's? Memory, learning, orientation, attention or concentration, executive function, and writing are impaired more. While in NPH, psychomotor slowing is there, and patient's fine motor speed and fine motor accuracy gets impaired. While motor and psychomotor skills, visuospatial skills, and language and reading are less impaired in Alzheimer's disease. While in normal pressure hydrocephalus, the memory, attention, executive function, behavioral changes occur more. Both causes impairment in memory. Alzheimer's disease had cortical abnormalities like aphasias, apraxias, agnosias, and impaired recognition and coding deficits. While the normal pressure hydrocephalus, there are subcortical abnormalities. Memory get impaired more, than slowing of the performing task and difficulty in completing the complex task. If we'll check with Parkinson, what happened? The NPH and Parkinson both have gait disturbances. Both patients take small steps. This and broad-based small steps. As the Parkinson increases, the step size get decreased. And in this, the patients have hypokinesia, freezing, imbalance, and extrapyramidal symptoms. A trial of levodopa can distinguish in between Parkinson's and normal pressure hydrocephalus. So how do we investigate the patients of normal pressure hydrocephalus? So when a patient comes to the clinic, what we do is we perform a CT scan as a baseline or a MRI. But CT scan we prefer as a baseline. So we check the Evans index and we check the bicoordinate ratio, temporal horn enlargement, periventricular enlargement, cerebral aqueduct or forced ventricle flow voids, and then we also do radionuclide scans in which there is decreased clearance of the drug. And then these days another MRI which is being used, a sequence of MRI that is signed and in which shows the ventricular flow rate. So as the CSF production is more, so there will be more increased ventricular flow rate. So it is much more evident in sign MRI, which is very much specific and sensitive for the normal pressure hydrocephalus. And then spectroscopy with AZM to check decreased paraventricular lucency, which is not reversed with AZM alone. So we will see it as we see the image. So what each of them. So if we see this MRI, so what happens is this: when a patient of hydrocephalus comes, we see 4 or 5 things at first. One, either the sulci are present or not. In the aging, the sulci are more increased, and in usually the young patients, they are not that much prominent. If there is hydrocephalus, usually what happens? Sulci is effaced. But in normal pressure hydrocephalus, for example, we see this patient, what we see that they are prominent Sylvian fissure and sulci are much more evident. Then what we do, this is the coronal section basically, and we see that the frontal horns are dilated, appearing as Mickey Mouse ears. And then we see third ventricle below it, it's also ballooned. And if we see downwards a bit, the horn shape, these are the temporal horns, and temporal horns are also dilated. So temporal horns dilated, third ventricular dilated, frontal horns dilated, sulci are more prominent, showing the hydrocephalus, but it is compensatory. Otherwise, if the hydrocephalus is acute due to any reason, what happens? Sulci get effaced. So with the age, as the aging occurs, our sulci brain gets atrophied, sulci gets more prominent. And when the sulci gets more prominent, what happens? The CSF gets accumulated in it. So we check the callosal angle. Callosal angle is basically we see the distance and angle between the corpus callosum and 2 horns, and it is really greater than 120. If it is So then it means the patient had hydrocephalus, and that is of normal pressure. Then another one is bicoordinate ratio, which we discussed in our previous slide. We take the we take an imaginary line, we cross it, and we check it crossing the coordinate nuclei, and through this we check the distance. Then Evans ratio, and in Evans ratio we also check the biparietal diameter as well. In the Evans ratio, what we do is we check the internal diameter of frontal horns, then we check the biparietal diameter, we divide it, and if it's less than 0.3, it's significant. So this is checked. So otherwise, if we see that the ventricles are grossly enlarged and when sulci are present, so sulci basically gets effaced. It's like a balloon. If the balloon is being pushed, and then if the balloon gets filled with air or water, what happens? Its involutions gets decreased. So we can identify. The purpose of describing the scan more is basically when our neurology junior postgraduate residents or our students used to see the patients, they get very much confused in describing the scans. They confuse it with acute hydrocephalus. Even the neurology colleagues usually ask us for surgery, for shunting. So sometimes we refuse them back. So they ask questions, this question, that why the patients grossly, the ventricles are very much dilated, but why are you referring the patients back? So the reason is this, that it is compensatory dilation. So how will we check the prognosis? So what we do is, initially when the patients of normal pressure hydrocephalus comes to us, we suspect that it is a normal pressure, we perform a lumbar puncture. With lumbar puncture, usually what happens, if the symptoms, gait is improved, then we drain 40-50 cc lumbar CSF, and if the symptoms like gait is improved or incontinence is improved, we prefer this patient for shunt. Shunt means ventriculoperitoneal shunt. This is also a kind of implant whose one end is placed in the ventricle and the other is in the peritoneum through the subcutaneous tissue. We'll see it afterwards. There would be image, and so it is done. If the lumbar puncture is positive for the patient's symptoms are improved, then patient will get benefit. So it's a prognostic test. If not with this, patient's extended lumbar drainage usually done, or patients are given CSF outflow resistance is being checked. If it's greater than 18, patients will get improved. We already discussed sign MRI, and it describes about the ventricular flow rate. If there is increased flow, patients will likely get benefit. So diagnosing an NPH is basically divided in 3 categories: probable, possible, unlikely. If the patient's age is greater than 40, patients are symptomatic for more than 3 months, there is gait disorder mostly, patient has urinary incontinence or dementia. When we are doing lumbar puncture, the opening pressure is 70 to 245 mm water, and the Evans index is greater than 0.3. Patient's temporal horns are enlarged, and periventricular lucency is there, opacification or lucency, and aqueductal or forced ventricular flow or callosal angle is seen. So then patient is suggestive of NPH. One thing that I wanted to mention is periventricular lucency. Periventricular lucency is transependymal migration of CSF. What happens when the CSF is when there is acute hydrocephalus? If we see this frontal horn, what happens? The CSF get past it, and in this image, it is not much evident or not present exactly. You will see that there is a cellite. Compared to this, the adjacent region gets hypodensity. Hypodensity means slightly grayish like shadow. So this is called periventricular lucency, means seepage of water transependyma through transependyma. This is called periventricular lucency. If it is present, it is a feature of acute hydrocephalus. So what is unlikely in these patients? If the patient had papilloedema, other reasonable cause, triad of gait, Urinary incontinence or dementia is not present, it is unlikely that the patient had normal pressure hydrocephalus. Papilloedema is there. Go for another reason. A kind of obstructive hydrocephalus is there that is causing papilloedema. The middle category is possible. We can possibly think that the patient had a normal pressure hydrocephalus. When the age is a bit less, symptoms are also less. Symptoms are means plus and minus. Means the patient had some symptoms or not. CT gets abnormal and lumbar drainage is also abnormal, but not that significant. So patient, we can suspiciously think of it, but the criteria which we match is that is probable for the diagnosing the normal pressure hydrocephalus. So this is a tape test. In the tape test, what we do is, as we mentioned, we put a lumbar puncture. We do a lumbar puncture, pass a LP needle, and passing the LP needle, we will drain around 40-50 cc CSF. So passing the 40-50 cc CSF, what happens? We see afterwards the patient get improved or not. If the patient's gait or incontinence get improved, we will consider the patient good for shunting. Otherwise, patient is not good for shunting. The other thing is we will do an external lumbar drainage, and in that we can put a tube connected with the with this needle. To needle is passed, and we put a lumbar drain, and then we will see it as it flows in the urine, as it flows in the drain bag. So if the symptoms get improved, again we will go for a shunt. So what is the treatment? The treatment is ventriculoperitoneal shunting, ventriculoatrial shunting, any kind of shunting. Shunting means giving bypass to CSF pathways. Other is endoscopic ventriculostomy. These both procedures are performed in our setups, and in most of setups, these are being performed. Endoscopic ventriculostomy trials are being done. It is not. It is a relative indication to perform a kind of procedure for a normal pressure hydrocephalus, but shunting. In this, the shunt which we use is basically programmable shunt. What happens that the shunts which are being used for this, if you go, these are programmable shunts. If we see, so what happens? These are chambers of these shunts. In this, the patient is already programmed regarding CSF pressure. So these programs, they can be programmed that how much pressure we want to develop. So if the patient's pressure gets increased, then that the shunt will work, and it will bypass the CSF. Means that if it is greater than 18, it will work. If it is less than 18, it will not work, like this. And otherwise, if but the issue is, again, this is cost effective. This is cost. The cost is much more. It costs around more than 1 lakh. The shunt is programmable shunt. So in setups like our setups, in government setups, what we do, we also put shunts, high pressure shunts. So they are being used, but the ideal situation is this. The other is in the modern world, they are using different valves, G valves and delta valves for this as well. Sorry. So this is the post-shunted video of the same patient, which we saw initially, who had difficulty in gait, magnetic gait. He had magnetic. Now, if we see the patient's walk, so much improved as compared to initially. He is turning good and taking the steps much more easily and smoothly. So what are the complications related to it? There is 1% chance of mortality. Infection regarding any foreign body, when we implant any foreign body in the body, what happens? There's chances of infection, and the infection chances are 3%. As the CSF pressure gets reduced, there are chances of, again, subdural vessels get bleed and causing subdural hematoma. There are 12% chances of it. shunt infection, which is 6.7% chances. And sometimes there is malfunctioning of shunt, so we will do the reshunting, and it is the main complication basically. 33% times the patients get shunted again in during their life. So how do we decide whether to shunt or not? So patients with high CSF pressure should be promptly investigated. If the response to 40 to 50 ml of taping or doing a tape test, lumbar tape, is there and patients gets benefit, it will respond to shunting. If we'll do external lumbar drainage, patients are evaluated, and if the patients respond, then the chances are better. If there is no substantial predictive value to MRI CSF flow studies, there will be less chances to get benefit. If multi-infarcts or Alzheimer's disease or dementia, there are less chances that the patient gets improved with the shunting. So those who gets benefit or good response to shunting are the patients whose clinical presentation usually match, as we have already discussed regarding age, symptomatology, and then tried everything. The gait disturbance usually presents first, as we already mentioned, then mental impairment, then the short duration of mild mental impairment. Patient had any known cause of normal pressure hydrocephalus like infection or bleed. So those patients who respond good to shunting are especially when there is lack of white matter lesioning on MRI, or the patient's symptom with CSF drainage improve. Patient's EEG is recorded. In the EEG electroencephalogram, what beta waves are greater than 50% with continuous intracranial pressure monitoring, then these patients get benefit. Resistant to CSF outflow if it is greater than 18 millimeter mercury. Are benefits of shunting long lasting? Most studies have shown fairly significant decline with time. Initially, the patients get improved. 60 to 75% of patients get improved initially. Afterwards, there is sustained improvement in only 24 to 40% patients. Results are confounded due to high mortality from comorbid conditions. 50%, 57% of patients die within 5 years in a study, which is given by Reftopoulos et al. So as the age is increased, patients had comorbid, so patients usually die because of other things as well. So the follow-ups usually are not that good. Those who are poor respondents are patients with severe dementia, the mental deterioration. Patients with mental deterioration will respond less. Dementia, if it is a presenting symptom, and then patients present with other symptoms, go for other diseases. But if the patients had initially the urinary incontinence or gait problems, they may get improved more better. Then we already read the MRI abnormalities. We saw atrophy of cerebellum, multiple white matter lesions, etc. Then these patients will have other causes of these issues, these problems, causing these problems. That's why it's less chances to get improved. So what are the recent advances in this? Recent advances related to this procedure? There are adjustable shunt valves that are adjusted with the pressure, opening pressure. We mentioned that we will require programmable shunt, and we will program it accordingly, whatever we need the pressure. And then the gravity controlled valves, low valve opening pressures when the patient is lying down, that is called G valves. They have low risk of over drainage. One thing is that in the routine, which other kinds of shunt except the programmable shunts, which, as I mentioned, that sometimes we use in the government setup because of the lack of resources. So shunts which are used are being middle to low pressure and high pressure shunts are used, but there are chances of over drainage because it is not programmed. It will drain all the time. So in the programmable shunt, when the pressure is not the exact, it stops working. Then it automatically starts working when the pressure gets increased. So these are the studies which show the pooled mean response rate of shunting in normal pressure was 59% in a meta-analysis by Heb. In those with good long-term survival, sustained improvement is possible with a rate of 39% documented after 5 years. So what's happening and where it's happening in Pakistan? So in Pakistan, Karachi as well as in Lahore, these are the two areas where these procedures, dystonias, Parkinson's, tremors, all are being treated. And Lahore was the initial setup where it happens. On my left is Professor Khalid Mahmood Saab. So he is basically the head and director of Punjab Institute of Neurosciences, Pakistan Institute of Neurosciences, PINS, and in that this is 500 bedded hospital at Lahore, in which there are 3 units for neurosurgery dedicated to neurosurgery. In this, he was a pioneer of DBS and all these in Pakistan. In Karachi, in the middle with beard, there is Professor Abdul Sattar Hashim Saab. He is doing here these procedures in Sindh. He is the only person in Sindh who is doing these procedures, and he is doing it in neurocancer center where these gamma knives and other things are also being done. And the right is Professor Asif Bashir on the extreme right, and in between is Professor Shehzad Shams. Both, one is at King Edward Medical College and Mayo Hospital, and other is again in PINS. So these 4 gentlemen are doing these procedures in Pakistan, at Lahore, and in Karachi. As we mentioned, that if the patients are there, we can refer them to Karachi Neurocancer Center Institute or PINS, Lahore. So now coming towards a small quiz, what we discussed. So anybody who is interested can go for it. If we'll stimulate subthalamic nuclei or globus pallidus interna and thalamus, what would we response? So this is what we already discussed. So now this is basically a tutorial. So if anybody is interested, he can answer. Anyone? No one will answer. Okay. So as we mentioned that all kind of rigidity, dyskinesias, tremors, all get improved with stimulation of subthalamic nuclei or globus pallidus interna. While in Parkinson, that gets less improvement if we'll target the thalamus, but the tremors are much more improved if we'll target thalamus. Other question regarding normal pressure hydrocephalus, how to identify which patient will improve after shunting. One of the most important cause we discussed, that which symptom if presents earlier, the patients will get improved with shunting. Anybody? Okay. If the patients present with gait disability or urinary incontinence, these patients are likely to improve with normal pressure hydrocephalus rather than the patients who present with dementia. Those patients with dementia are less likely, there are less likely chances that the patient will get improved with shunting. How can we label normal pressure hydrocephalus on MRI? So what is happening okay, we will see these questions and answer afterward, but in the end, so how can we label normal pressure hydrocephalus on an MRI? So on MRI, as we mentioned, we will check the callosal angle, we will perform sine MRI, we will check the Evans index, we will do pi-coordinate index, we will check the biparietal diameter as well, and then we will identify along with it if the horns are dilated, temporal and frontal, third ventricle is dilated, the seepage is there, periventricular lucency or not present or not, and then we will see the sulci as well. Sulci gets prominent. So with checking these, we can identify that the patient is suffering from NPH or not. Why it is not working? So what is the target used in treating the dystonias? So these are basically globus pallidus internus. A lot of questions. So we will try to answer these. So thank you so much. Jazakallah. This was all about neurosurgery and its role in neurodegenerative diseases. This is our ward and our team, along with me, our chairman, Professor Riaz Ahmed Raja, fellow colleague Dr. Sanaullah, Dr. Vashdev, and other team is standing performing a surgery. So now I'll try to answer your questions. So are there chances of hardware complication like eroded lead wire? Yes. There are chances like shunt. When we perform a shunt, similarly, when we are implanting a tube, for example, DBS tube, there are chances of erosion. If we'll press, if we'll scratch, we will get the skin gets infected. There are chances that it gets eroded. And DBS is effective for Parkinson patient. Yes, it is very effective. It is very effective, as we have seen the videos. Actually, in our center, we do not perform, but as I mentioned you that it is performed in 2 of the centers of Pakistan, one at Karachi and one at Lahore. So 2 at Lahore, one at Mayo Hospital and other is PINS. PINS is the largest center. So if you will come to counter their videos, they have a lot of videos as well. Patients get treated, our patients treated, but as it is unethical to take their videos, so I took the internet sources and show those videos. What symptoms of Parkinson's disease get most benefit with DBS? So tremors improved a lot. Then bradykinesias. Tremors are those which improved a lot. Although electrical impulses from the DBS block signals and scissors, but it's hardly performed in majority cases in Pakistan. It is due to the system expensive. Yes, Ina asked that the system is very expensive. Yes, you are very right, Ina. The system is so expensive that it is not performed. Like in our setup, the government is approved and we are waiting for it for last 1 year. Inshallah, we will get it and soon we will also perform it. But again, as our country, there is a lot of poverty, so not every patient can get benefit from it. Patients can die from DBS surgery, definitely. If you will not target properly, you will get, and if you will not plan it properly, you can hit the vessels. Patients can get the intracerebral hemorrhage, intraparenchymal hemorrhage. Patient can die of it. Yes, batteries get changed, and I am not exactly sure that how expensive different varieties are, but any category more these Meditronic and one more company is providing these. Both are equally expensive for our people. Why would DBS use a subthalamic? Both the subthalamic nuclei and globus pallidus internus can give us better results, not a single. Both with using both, we can get the same results. What concerns Alzheimer's shunt patients? Farah Batul asked question that the Parkinson, Alzheimer's, and shunt patients. I do not got the question regarding.
Okay, so would that be humble if you please unshare your screen for now?
Sorry,
if you can unshare your screen recording.
I did not record it.
Okay, so there is a question from our viewer from the YouTube. Her name is Sarah Haki. She's asking, what about the tics old people develop without other motor disturbances? Is DBS effective for that?
Yes, DBS is effective for it, and patients get the patients with tics get treated with the DBS. But again, with it, the patients would undergo the different performance evaluations. That either the patient is exactly the super selected, means the patient gets super selected, that the patient will get benefit or not. But DBS is being done for it as well.
So another question we have is from Shuja Parkinson's or any other neurodegenerative disease?
Yes, DBS is being done for so many diseases. Like its role is in multiple sclerosis. Patients are. It is also being done for cerebral palsy and other diseases. So deep and along with the deep brain stimulation, the stimulation techniques are being used. Spinal cord stimulation is being used for spinal problems, but regarding neurodegenerative disease as a topic of our focus on these few diseases, DBS is being used.
Okay. Rushda Afroz asked us, while placing the electrode for DBS, is there any chance of damaging or injuring nearby regions?
Yes, there are high chances of damaging the nearby structure. That is why, if you remember, I showed a video in which there was precisely being electrode was being placed. That is a frame through stereotactic frame. What we do is we pinpoint exactly with using the MRI neuronavigated. We had minimum margin of error is less than 1.5 to 1 millimeter. We cannot go beyond it. Otherwise, you will damage other things and patient will suffer.
Okay. And is there any chance the patient can develop any type of complication after the DBS surgery?
There are a lot of complications. With every surgical procedure, there are chances of different complications. So if you will not, again, the point is, if you plan properly, you will get less chances of complication. If you will not, patients can have problem with speech, patient can have problem with dystonias, again develop dystonias or movement disorders, patient can get blindness, patient can have intraparenchymal hemorrhage, a lot of problems, seizures, many problems. So it's all it's all basically the operator dependent, how experienced you are and how precisely you perform.
Okay. Another viewer from the YouTube, Fawzia Batul, asked us that if a patient does not want to go for the lumbar puncture, what can be the alternative or the best option that?
Basically, what we do is lumbar puncture. In our setups, usually patients do not want it to are afraid of doing a lumbar puncture, but it's very easy and it's very good, and the results are very satisfying. So we always prefer it. But for example, like the sister asked, that if the patient doesn't want to go, then we can do a frontal burr and we can check pressure from there as well, and we can go means from frontal region we can do it. We have to measure the ICP. Otherwise, if you will go directly for shunting, there are chances that you can fail. Sine MRI is also helpful, very helpful. Sine MRI. So in sine MRI, if we see that there is a flow rate, ventricular flow rate, with it, along with it, if the patient's EEG, if we record beta waves are there, and ICP measuring, if these things are being checked, one can get, if like the sister asked that someone doesn't want to get lumbar puncture, then these things can be checked, and if these things are checked and these things suggest normal pressure hydrocephalus, then patients can get benefit as
well. Thank you so much, Dr. Asad. We are ending up here with the question answer session. Off to Dr. Sadaf.
Thank you so much. Jazakallah.
Thank you, Dr. Pirasat, for such a elaborative session. And we are very much proud that you are still the most well-learned person in the team related to this point and related to this theme. So that's why I requested you to just present and highlighted these areas. And most of the participants are from the basic sciences, so they were very much interested in knowing about the clinical side and these things. Most probably, we will also be having some queries in next few days as well for you. So we will keep you posted. And if you can answer, so we can take you in on the Zoom session in the same ongoing session during that, or we can send it to you via email, so you can send the answers. Because people are very excited to ask some related topics.
Definitely, they all are welcome, and it will be a pleasure and it will be honor to answer them, to help our fellow colleagues. Definitely, as this is Basic Science Conference, First of all, it's very good to have such kind of atmosphere and such kind of confidence under the shadow of neuroscience, all the fields, the clinical, which we are a bit far from the basic science, we get closer, much more closer, and we can treat the patient
exactly. Exactly, because all the basic scientists are actually looking for the cures of certain diseases and doing research for their causative agents and everything, but there is no gap linkage between these two entities. So in our country, this is much more a dilemma, and we are like people like you and Dr. Anam are actually connecting these things. But thank you so much for this enlightening session, and I hope people will enjoy. We will shortly share the feedback with you as well.
Thank you so much. Jazakallah. Thank you so much all. Jazakallah.
Thank you. Thank you. Thank you. Hafiz. May I announce it? Thank you so much, all the participants, and we would like to see you tomorrow with the talk from Professor Dr. Jerry DeVore, and he will be talking about all the psychophysiological aspects of the recordings and the analysis and the assessment of neurodegenerative disorders. Dr. Jerry DeVore is a very well-learned professor, a very senior professor of psychophysiology, and beside being a very great time zone and the whole day and night duration difference, he will be there for the question answer session, though he has sent his video talk, but he will be there live for the question answer session. After that, we will be having Professor Dr. Tawqir Ahmed from NUST. He will be sharing his studies on mice model he has done for the memory deficits and dementia related research. And followed by that, we will be having Professor Dr. Ubaid Ali, who has association with Ministry of Health and Drug Regulatory Authority, Pakistan. He will be talking about some novel treatments in the market and in the upcoming pipeline and the future strategies to treat the Alzheimer's, Parkinson's and other neurodegenerative disorders with the perspective of pharmaceutical industry and pharmaceutical sciences. So, and then followed by a lab tutorial, very exciting lab tutorial that how you can prepare the animal models for the neurodegenerative disorders by Dr. Ruxana Nawaz from Alan University, UAE, and Dr. Saima Gul from Shifa Tamir Millat University, Islamabad. So we have whole packed jam session and all the live talks for you tomorrow. So looking forward to meet you all with some very related and very thought-provoking questions. Thank you so much.